I mean, they would move it. I know, but... And I'm assuming everybody can see my screen, otherwise somebody would have mentioned that they don't see it. So, okay. And please mute yourself if you haven't done so already. Okay, so last time we talked about, I'm just gonna go a couple of slides back to kind of highlight some of the things we discussed or finish up with last time. Guys, you need to mute yourself, please. So when you're looking at the process of movement across the plasma membrane, one of the first things that I mentioned to you guys is the process that we're discussing takes energy, yes or no? And we said if that process doesn't take energy, we refer to it as the passive process. And if it does require some sort of energy, which is typically ATP, then that would be referred to as an active process. And as far as we got last time was looking at the three types of passive processes, which were basically diffusion, when the molecules are small enough, I guess as I said, diffusion simply meant that molecules move from high concentration to low concentration. But if I'm using the word simple diffusion, that simply means, use of the word, that simply means that the molecules are small enough and that they can just sip through the plasma membrane on their own. They don't need any help from anything. When we're talking about the second type, which is, and I said just simplify it, we're just gonna call them facilitated diffusion, is that these molecules still can pass through the membrane, and again, it can be either direction. They still don't need energy. However, the word facilitated implies that they do need the input of the transport proteins or membrane proteins to get in and out. So the facilitation comes from the membrane proteins. And then lastly, what we talked about was osmosis. Osmosis was basically only and only diffusion of water. It's limited to water only. So make sure you guys remember that in terms of water. So please, please do not use the word osmosis and diffusion interchangeably. You can say water diffuses, but you cannot say that any other molecule uses osmosis. So the word osmosis is specific to your water molecules only. And I usually have a question about aquaporins. So again, remember aqua means water and porin means pores. So aquaporin basically means pores of water. It allows the water to get in and out. And again, if you're just walking in, please mute yourself and make sure you type your name in the chat box. So what we're studying right now is basically the new information associated with active transport. And again, as soon as you hear the word active, guys, you should automatically go to the fact that this process requires some sort of energy. It doesn't matter what type of energy we are. We're not going to go into that detail for this class, but it needs some sort of energy. So if you look at this, there's two main types of active process. One is that is just referred to as an active transport, and the other one is referred to as the vesicular transport. So active transport itself is divided to two types, primary and secondary active transport. So Primary active transport, I'm just going to move on to the next slide so I can show it to you. Primary active transport implies that the main way, again, active implies it needs some sort of energy. But when I use the word primary, it implies that the main source of energy for that movement is going to be ATP. So if I want to move a molecule, I must have ATP present. That's all you need to know when I use the word primary active transport. That is all you need to know in terms of what that means. It means, again, it requires ATP to move the molecules. Now, the second part of this is the example that I provided. And this example is going to haunt you in physiology, guys. So learn it now and make sure you guys are comfortable with this. So primary active transport, the best example we have for it is a pump called sodium potassium ATPase. And notice the word ATP is already built into that, which means it requires that ATP. So what does this pump do? And it's demonstrated in a picture and you can take a look yourself. Every time this pump uses an ATP, one single ATP, it's going to move 3 sodium outside the cell and 2 potassium into the cell. So that movement has to happen when an sodium potassium ATPase is used. And the reason I'm saying it's so important is because literally the function of your cell, the function of your, you being able to live, your neurons to work, your heart to pump, your muscles to contract is sodium potassium ATPase. If that channel has problems, then you'll die. It's as simple as that. And you die in a matter of seconds. So sodium potassium ATPase is extremely important. And again, it is typically the best example we use for primary active transport. Now, the secondary active transport we have, which is listed here, and I'm not asking you to go into details about this, is that anytime you hear the word secondary, it implies that it still uses energy, but that energy cannot be ATP. So it's using some other source of energy, okay? And again, because it's the word active, it means that it's moving molecules from a low concentration toward the high concentration. So it's kind of like, think about it, moving a stone up a mountain, right? So if you're pushing a stone up the mountain, you as a person are putting effort into this, you're putting energy into that. So same concept applies here. So secondary active transport implies that you're moving the molecules or the components from a low concentration to a high concentration. But the word secondary implies that it does not use ATP, it uses some other source of energy. Now, for our purpose, I'm not going to go into details about this. All I want you guys to know is that there are 2 types of secondary active transport called antiporters and symporters. And that's all you need to know. That's the amount of detail I want you to know. Nothing beyond that. Are you with me so far? Is there any questions about this? Okay. Now, vesicular transport, which is another form of active transport, is when you are going to move molecules that are typically large in or out of your cell. So large molecules getting in and out of your cell. Now, these molecules cannot simply pass through your plasma membrane. So what you need to do is literally kind of have those molecules, if you want to bring them into the cell, have them in this, and I'll show you a picture because I can use my hands very effectively. So let's say this is my cell, and I'll just show you this. Here's my cell, and I want to bring this particle in, okay? So when I want to bring this particle in, this particle is large enough, right? So I can just bring it in through the plasma membrane. So what is my solution? So see how the membrane is creating this invagination, this dip inside of it? And then eventually membrane is gonna wrap itself around that particle. So it's not that the particle directly comes into the membrane. It's like literally you bring the particle into this space and then you close around it, and then you bring it into your cell. So the new thing that came in, it has the plasma membrane wrapped around it, and it has the particle at its center. So what we brought in is called a vesicle. So vesicle, not phagocytic vesicle, that's a specific to this case, but just vesicle. Now listen to this word, guys. Look at the word, guys. Endocytosis. What is the word endo means? Anybody? Anybody knows what endo means? Endo versus exo. Endo means inside, correct. So endo inside, so exo means outside, right? So kind of like opposite of each. So exiting, kind of like I always think about exo as an exit. So what is the word site means? This we talked about last time or a couple of times ago. Cells, correct. So when you see endocytosis, what does that mean? Bring the word together, guys. Remember, anatomy is a combination of multiple words together. So endocytosis means you are bringing something inside the cell, correct, you got it. And then exocytosis literally means what? Taking things out of the cell, correct. It's literally the opposite. So look at the picture, guys. Here is a vesicle that I have right here. So the vesicle is gonna come toward the membrane. So here's my vesicle right here inside the cell. It's gonna come up, join the membrane, and as soon as it joins the membrane, it opens up to the outside and literally dumps up its particles to the outside environment. So endocytosis, bringing something in, exocytosis, taking something out of the cell. And in both of these scenarios, you're using energy in the form of ATP. I think somebody asked me a question. So yes, so the question is, so does this work the same way as when pathogens are present? So the process of endocytosis and exocytosis is used for movement of large molecules. If we specifically talking about white blood cells, which are responsible for the defense of your body, then pathogens are basically anything that can cause infection, like bacteria or viruses. They can be brought into the white blood cell, chopped up to little pieces, and then basically the waste product of it gets exocytosed. But yes, you're correct. But is it limited to pathogens? No, it is not limited to pathogens. It's for all of the cells that are basically alive and can carry endocytosis. Okay, so that's as far as the membrane transport goes. The process is the same. The process is identical regardless if we're talking about exocytosis or endocytosis. It's just when we're talking about specifically the white blood cells, we call those phagocyte and phagocytic cells. Okay, so when we're talking about your cell, we talked about the plasma membrane, which is something we focus on so far. We talked about how things get in and out of this plasma membrane, which is either through a passive process or through an active process. And we've kind of looked through examples of those. Now we're going to take a look at what's happening inside our cell. So we're going to start with cytoplasm. So cytoplasm is literally the space that is inside the cell. Okay. So what is it made up of? What is the cytoplasm made up of? It's composed of this gel-like material that is literally filling up the cytoplasm. So cytoplasm is the actual space, if you want to think about it. And if I fill that space with this gel-like material, that gel-like material would be called cytosol. Now, inside this cytosol, inside this gel-like material, I have things called inclusions, which are basically molecules that are kind of floating inside the cell. Your cell needs them to survive. These are like glycogen, like basically carbs, pigments in terms of color pigments, lipid droplets, which are basically fat, vacuoles, which are usually filled with fluid, and then crystals, which are not really common, but they can be seen in some cells. And then you have organelles. This is what we're gonna focus on, guys. We're not really going to go into inclusions. We're gonna focus on organelles. Now look at the word, guys, organelle. Organelle means literally little organs. So if you think about the organs that are in our body, each of the organs in our body do a specific job, right? Their job is, let's say your heart is to pump, your lungs is to breathe, your I don't know, your liver is responsible for production of bile and detoxification, etc. So the organelles that you have in your body do the same thing in terms of they are much smaller, but each type of organelle has a very specific function in our body. So what we're gonna do today for the rest of this lecture is learn what are these organelles and what is the function of each of the organelles. Now, one of the things that I've listed here, and usually this is when I have plenty of time, which is most of the time not the case, is we look at organelles in terms of if they have a membrane around them or they lack membrane. For our purpose, for our lecture, I don't need you guys to know that fact. So I don't need you guys to distinguish between membranous or non-membranous organelles. As far as you know, it is an organelle and it does this whatever function in your cell. So that's the focus for us, okay? So ignore what in terms of if the membrane is present or not. I'm not gonna ask you questions about that. I'm not gonna ask you guys to distinguish those. So as far as you know, the name of the organelle and the function of it, you're good. So here I separated them again, but we're just gonna look at all of them without that distinguishing fact. There you go. So let's just start with, even before we go into our organelles, let's talk about nucleus. Nucleus is technically not an organelle, even though I said it is organelle, but in many textbooks, if you read it, and I think in your actual anatomy textbook, it refers to it as organelles and then nucleus. So it doesn't really refer to nucleus as an organelle. But again, it depends on which textbook you read. So nucleus, for our purpose, and what I want to teach you right now, we're going to say it is an organelle. It is the largest we have inside the cell. And basically, this is what you see. So if this is my cell, this large purple circle you see, that's your nucleus, okay? Now, we already kind of learned about nucleus last time, right? Because you guys did the mitosis lab. But just kind of going over some of the new vocabulary that you might have not heard. We have the membrane envelope, which is called the nuclear envelope, that surrounds your nucleus. And the way I described this to you guys last time was I kind of remember that nucleus contains your DNA. And because DNA is so important, you want to wrap your nuclear envelope with this kind of barbed wire protective shell. And that's your nuclear envelope. Now, nuclear envelope still needs to be able to communicate with the external environment. Now, how the external environment of the cell, not outside the cell. So the way it does that is that nuclear envelope has these little tiny holes inside of it that are called nuclear pores. And that's basically allows things like RNA to exit your nucleus and be used to make what? So what is the purpose of RNA, guys? Why do I need RNA? To make protein, correct. So it's extremely important for me to be able to access outside the cell to transport things like RNA to the outside of the cell. The other things that I have is called a nucleolus, not to be mistaken with nucleus. So nucleus and nucleolus. So if you look at the picture we have here, the center of your nucleus, you have this blue circle, and this blue structure is your nucleolus. What is the importance of it? It's where your ribosome manufactures. So I'll talk about ribosome in just a minute, but it's important. So if I say, where is the ribosome actually coming from? It's coming from the nucleolus, which is inside the nucleus. So be careful with the vocabulary. And then lastly, we talked about our DNA. We said, again, DNA is protected inside the nucleus. So we just don't want it to have it as a really long strings of DNA because it is fragile and it can be broken down. So what I do to that DNA is I'm gonna wrap it around proteins, and that is going to create something called a chromatin. So again, this should not be a new word to you since we talked about this last time in the mitosis process. So here's our picture again. These little orange things that you see, those are your pores. The purple lining is your nuclear envelope. The center is nucleolus. Now, what you also see in this picture is that other than this purple blob, you also have these kind of extensions that are coming off in color of blue with these little yellow, not yellow, sorry, apparently I'm colorblind. These orange circles right on top of it. So that's the part that we're gonna talk about next. So these folding of membranes that you see right here are referred to as, and I'm gonna skip a couple slides and come back to it. It is referred to as endoplasmic reticulum. Endoplasmic reticulum, which is simply, in many cases, I just simply refer to it as ER, okay? So again, these are membranes that are kind of stacked on top of each other, and they are the continuation of my nuclear envelope, as you can see in this picture again. So here's my nuclear envelope, and this is the continuation of it. So if this membrane, if this endoplasmic reticulum that I'm talking about, if it has ribosomes on its surface, remember ribosomes that I said it's made inside the nucleolus? If I have those ribosomes present inside, then they appear as like these tiny dots on the surface of the endoplasmic reticulum, and it creates a roughened surface on the ER. Therefore, we refer to this as rough ER. Now, if part of this ER does not have ribosomes attached to it, these tiny dots that I showed you in orange, if these dots are absent, then the surface of the ER would be smooth, and I literally refer to it as a smooth ER. So let's take a look at the picture that I'm talking about. So look at this again. The blue represents the ER. If you have these tiny orange dots of ribosomes on the surface, this portion is called rough ER, and the part that is missing those is referred to as the smooth ER. Is this making sense so far, guys? Any questions, any clarification? Awesome. Okay, perfect. So we're talking about rough ER and smooth ER, but before we go in, let me just go back to what ribosomes is so we can connect the ribosomes to your endoplasmic reticulum or your ER. So ribosomes are basically made up of what we call ribosomal RNA or rRNA and protein. And again, if I ask you guys the question, where is ribosome synthesized? Where is the ribosome itself synthesized? What would you say? Nucleolus, correct. So nucleolus is where you're actually synthesizing your own rRNA. I was like, what is this called? rRNA for the purpose of creating a ribosome. Now, I don't want you guys to know the detail about how that process happens. Again, all we just want to know is what is the ribosome, where is it created, and then what is its function. So what is these ribosomes, these tiny orange dots that I showed you, actually do in our body? And their job is to what? Synthesize protein for us. So let's bring these concepts together, guys. DNA is found inside, I'm gonna just focus on this guy. DNA is found inside my nucleus. The components of RNA is found inside the nucleolus. So I produce RNA and I kick it outside into the cytoplasm. Then I take my DNA and I make copies of this, which is called RNA. And I kick the RNA out as well. Now I have the RNA and the ribosomes coming together, and with the help of both of these components, I'm going to produce proteins. And proteins, again, basically, as if you guys remember from our last lecture, they basically do everything in our body, including chemical reactions, breaking things apart, forming larger molecules, etc. Now, the ribosomes that we're talking about, which are basically responsible for protein synthesis, can just be moving around the cell. They don't need any help from anything. They just look for RNA and they make a new protein. That's it. That's what they do. And they're called free ribosomes. Kind of makes sense, right? They just kind of look around. I have a job here. Okay, I get it. I have a job there. I have a job there, right? So they're kind of like consultant, if you want to think about it. They're free floating. If they are membrane-bound ribosomes, they have to be attached to your endoplasmic reticulum. And remember, if ribosome is attached to your endoplasmic reticulum or your ER, that makes your ER a rough ER, right? Because they mix those bumps on the surface, make the surface rough. So we have a rough ER, we have a smooth ER, and the only difference is in terms of a structure is that one has ribosomes attached, the other one doesn't have anything attached to it. So how does that affect the function of the rough ER versus the function of the smooth ER? So rough ER is responsible for basically taking the protein that's already been created and folding it. Remember, when a protein is made, all it is a long chain of amino acids. There is no shape to it. So creating that beautiful 3D structure that we talked about it, that gives it its function, comes from your rough ER. The folding of it to create the 3D structure is coming from rough ER. But the protein that the rough ER just basically folded, it's not done. It's not ready to be used. So what it needs to go, it needs to go to another structure called the Golgi Apparatus. So what does your rough ER do? Your rough ER says, here's a protein, I folded it, now I'm gonna package it and send it to the Golgi Apparatus. So it's literally you delivering it from the rough ER toward the Golgi Apparatus. So if I ask you what's the most important job, and again, these are things that you want to review for exam. What is the most important job of your rough ER? One is folding your protein, right, as it's being synthesized. And then the second part is basically packaging that and sending it to the Golgi apparatus for final modification. Could ribosomes attach to the other organelles? No, they cannot. So ribosomes only either are free floating or they are attached directly into the ER. So somebody just, okay. So what is the smooth ER if it has no role in protein synthesis? So what do we need that for? So the main functions, and I listed quite like 4 functions, which is a lot for a relatively small organelle. So what does a smooth ER do? So a smooth ER is first of all synthesizing things like phospholipid, cholesterols, and carbs for the membrane. If you guys recall from our earlier part of the same lecture, phospholipid bilayer, remember that word? Which is your plasma membrane. So that phospholipid bilayer has to be created somehow. So where do I create it? Where is the manufacturer for that? The manufacturer would be your smooth ER. The other thing that it does, which the word should sound familiar to you, is taking down your glycogen. What is glycogen, guys? What's the proper, another way of saying glycogen? Or what category of macromolecules does it belong to? Let me ask you that question. Glycogen. Oh, guys, you're going to be having troubles if you can't answer that. Carbs. Glycogens are carbohydrates. They're complex carbohydrates that are found inside animals, right? They're not like a starch. So even if you kind of look at what I've written here, guys, you should be able to answer this. I'm telling you, it takes glycogen and does what? Convert it to glucose. What is glucose? Carbohydrates, right? So that should be already an answer for you. But going back, it says it's taking basically metabolizes lipids and break down glycogen and glucose. We're not really getting into this. That gets it a lot complicated. So the main thing I want you guys to know is conversion of glycogen to glucose when it's needed. It also detoxifies your anything that enters the cells, like things like carcinogens, things that cause cancer, pesticides, drugs, alcohol. So a smooth ER is really, really active in organs like liver and kidney, which kind of makes sense because liver and kidneys are the two organs that are used for detoxification in your body. And then inside a skeletal muscle specifically and cardiac muscle, they're responsible for storage of calcium. We're going to come back to this later on when we talk about your muscle contraction in the muscular system lecture. That is true. That is true. Sorry, I'm just reading the comments. So. Okay, so here's a picture again. We already seen this. So let's move on to the next structure. So if we're looking at the process that we described so far, we had Golgi apparatus. We had, I guess I shouldn't say Golgi apparatus. We had ribosomes that make the protein, and then your rough ER folded those proteins, and we said that was not done. That protein was not done. So it needed to go to the next structure, which is your Golgi apparatus. So Golgi apparatus, if you look at it structural wise, is somewhat similar to ER. So here it is, like see how the membranes are folded on top of each other? So it's the same concept. The job, however, is significantly different. So what Golgi apparatus does is that it takes the vesicles that are coming from rough ER and then it modifies this. So the modification is it adds tags to it, it adds glucose to it, it might add it adds lipids to it, it changes it so it is the final product basically. Okay, so brings it from the rough ER, modifies it, the final modification that protein needs, and then eventually when it's done, it packages it and sends it to its final destination, wherever that final destination is going to be. So a lot of times when I'm lecturing this, I'm telling you guys that the Golgi apparatus is like the FedEx. You bring a package to the FedEx, right? The FedEx does their own wrapping and make sure everything is secure, right? Packages it again in their own box and then deliver it to wherever it needs to go. So in terms of the Golgi apparatus, in terms of function, again, simplified, receives protein from the rough ER, modifies those proteins, and then send them to the final destination. Is this making sense so far, guys? Any question, clarification on anything? Okay, good example. I don't know what you mean by good example, but if you actually want an example, Oh, okay. Sorry. It's my pregnancy brain. Forgive me. Sometimes it just literally, I don't even know what I'm talking about. Sorry. Okay, so let's look at this as a whole scenario, guys, now. So nucleus made my DNA. Nucleolus made my ribosomes. They came outside, and thank you. They came outside, and the RNA and ribosomes together synthesized a new protein. Those synthesized proteins were sitting on surface of the rough ER. Rough ER folded the proteins, packaged them, and sends these packages to the next structure, which is Golgi Apparatus. Golgi Apparatus receives it, modifies it, and then packages it to send it to its final destination. So what is your final destination, guys? So that's the next question. So where is these proteins going to be used within my cell or within my organ? So their proteins have 3 destinations, guys. So pay attention to this again, typical exam question. One is that, or these proteins that are synthesized are just gonna stay inside the cell. So you can see it right here. They're not gonna go anywhere. They're just gonna stay inside the cell and used by the cell itself. The other ones that you have, they can actually get kicked out of the cell. So the proteins are basically used. They are not needed by the cell itself, but your cell needed to make it, and they get kicked out of the cell. And a really good example of this is hormones. When you make hormones in your body, the cell itself is not going to use it. It's going to release it outside the cell, which goes to your bloodstream. The last possible options is you take those proteins, and remember, part of your membrane are proteins that are built up inside your plasma membrane. So you can embed those proteins inside your plasma membrane. So again, 3 pathways. One, staying inside. The other one goes to the plasma membrane and gets embedded in the plasma membrane, or gets kicked out of the cells through the exocytosis process and used outside the cell. Different tissues. What do you mean by different tissues? If you can clarify that. Are you talking about, is this different for each different tissue? The answer is yes, this process is different. So 3 pathway, know those, make sure you can understand them, make sure you can distinguish them. And again, follow the path from all the way from DNA all the way to when that protein basically gets used inside the cell. Correct, yes. Okay, so the next organelle that we have is called mitochondria. So mitochondria is really, really important. And I think that's like among the organelles, it's my favorite organelle. So first of all, it is a double membrane organelle. So you can see this in terms of around the cell. That's one membrane. And then the internal membrane, which is this one. And of course, there's a gap between these membranes. Now, if I say, what is the function of mitochondria? The simplest way to say it is that it is the power plant of your cell. Now, what does that mean in terms of power plant? It means it is essential for synthesizing ATP, which we're all familiar with now, right? ATP production is responsible or is done by your mitochondria, which is something I mentioned previously as well. Now, this is really important, guys. What I'm describing in this process, in terms of mitochondria, it only does it when there is oxygen present. So it does that using a process called aerobic. Like, you know how aerobic means presence of oxygen? So if you're looking at an anaerobic process, which means lack of oxygen, AN and then aerobic, lack of oxygen, you don't need mitochondria. It can happen naturally, but it doesn't produce a lot of ATP. But when we're talking about 90% of the ATP that your body produces comes from mitochondria in the presence of oxygen, okay? And if that oxygen is absent, basically you lose the capacity to make significant amount of oxygen. If that problem with mitochondria persists, then you basically die because your body does not have enough capacity to produce ATP in the absence of oxygen. The other really cool fact about your mitochondria, it is the only organelle that carries the following. It has its own DNA, RNA, and ribosome. So if you have these three components, what is the capacity mitochondria gains, guys? What does mitochondria do that the other organelles that I mentioned are unable to do? Act as a cell is correct, but we're talking about organelles and a function. So a specific function, what can it do? Create protein or synthesize protein. You got it, guys. So basically, going to back what, sorry, I already moved from my thing. Swama says, you can take the mitochondria out of the cell and that mitochondria will be perfectly happy. It produces its own ATP, it has its own DNA, RNA, and ribosome, which means it can make its own proteins and live happily ever after. Can your cell live without a mitochondria? The answer is no, because if I take mitochondria away, what am I taking away from? What capacity am I taking away? Be careful, guys. It not just ATP, is the capacity to what? Make ATP, right? Because it's not the usage of ATP. So your answer needs to be %. So it loses the ability to make its own ATP or energy. So it's losing its power plant, basically. Okay. So the next organelle we have is lysosome, which are like these little tiny balls inside the cell, inside your cytoplasm. The characteristics of the lysosomes is that they are acidic and they are filled with lots of digestive enzymes. They're filled with a lot of digestive enzymes. So when they are filled with digestive enzymes, what they actually do is they're responsible for cleaning up large molecules that are in there. They digest pathogens. Pathogens, again, it's things like viruses or bacteria. They recycle if your organelles are damaged. They just don't let that organelle linger around. They literally break them down. And then if there is any waste product as a result of these breakdown, they literally spit them out of the cell. So they do that through the process of exocytosis. And we already learned about exocytosis. So I think about lysosome as your cleanup crew. They come in, if there is anything that needs to be cleaned up, it chopped it up to little pieces, and then the waste product gets kicked out of the cell. How does it accomplish this task? By having very acidic and highly enzymatic environment. The other really cool thing that lysosomes have the capacity to do is carry a process called autolysis. So I brought down the word for you. Auto means self, lysis means breakdown or breaking down. So let's say you have a cell and that cell is damaged. And the marking and the guideline inside the cell, it says, because I am damaged, I need to destroy myself. And this is a really cool fact about your body. So the way it goes about that, the way it thinks about this is that it literally allows the lysosomes to burst inside the cell. Remember, lysosomes are acidic and they have a lot of digestive enzymes. So when that the inside of your cell gets introduced to this really toxic environment of the lysosomes, it literally starts to be eaten up from inside. So your cell is literally self-destructing by breaking down lysosome and releasing all of those acidic environments into the cell. So I think it's fantastic, like way of your body just dealing with problems. If there is an issue with the cell, it tries to destroy it and get rid of that cell. Questions about that? Good. Okay. Preoxisome is another one, guys. Preoxisome is, a lot of my students confuse preoxisome versus lysosome. So please be careful with these. Preoxisome is associated with removing what we call metabolic waste, guys. So what we were mentioning earlier for the lysosome, they're just things that are products that are wasteful, like food particles or viruses or bacteria. They're not a result of the metabolism process. So what is the difference? So metabolism implies I am carrying up a chemical reaction, okay? So whenever I carry a chemical reaction, this is associated with your cell basically survival. One of the harmful things that is produced as a result of my cell just doing its normal day-to-day activity is something called hydrogen peroxide. So hydrogen peroxide is the waste product, okay? We also produce things called free radicals, which are basically these free unpaired electrons. Both of these can be really harmful to us and are harmful to us. So unpaired electrons can cause damage to your cell. It can cause cancer. And hydrogen peroxide are just simply bad for us. They can kill the cell. They can destroy the cell. So what is the solution for your cell to resolve this issue? So peroxisome, the organelle that we're talking about, contain two enzymes. These enzymes are oxidases and catalases. And what they do is they things that are harmful to us, like free radical and hydrogen peroxide, and convert it to something that has literally no problem and is actually good for us, which is water. So taking something that's really damaging to us and basically neutralizing it or simply getting rid of that toxin component is the job of peroxisome. I don't need you to know this because I really don't discuss it with you guys. But if I ask you what is the main function of peroxisome, this is what I'm looking for. It's getting rid of the byproduct of metabolism that are harmful to us, which includes free radicals and hydrogen peroxide. Last thing I want to talk about, and again, I'm going to simplify this for you guys, is what we call cytoskeleton. So cyto again means cell, and a skeleton means basically a skeleton. So these cytoskeleton, which are three types, and you can see them, they're just three levels of thickness. They basically form the shape of the cell and do major things in terms of transportation and motility inside the cell. So kind of think about them as the bones of your cells, not bones of your body, but bones of your cells. So again, there are three different thicknesses. The really smallest one is microfilaments. Somewhere in the middle, intermediate filament. And the one that kind of looks like a tube, which basically has an empty center, is called microtubules. Now, I don't need you guys to know a lot of detail about them, other than some of the main functions of these three cytoskeleton. And I'm gonna highlight those for you. So the thinnest among these cytoskeleton, as I mentioned, is microfilaments, which is about 7 nanometers, which is really small. It's the diameter of a typical red blood cell. So it's important in, I guess, 3 senses. Cell motility, so movement of your cell, changing your cell shape, so if it needs to stretch one way or kind of shrink from the other way, so shape of the cell, and then carrying out process like endocytosis and exocytosis. That's these type of microfilaments are involved in this process. If it becomes somewhere in the middle, which is your intermediate filaments are a little bit more involved in the process of the cell. So what they do is they stabilize your organelles. So they make sure basically that the organelles are localized in their position. They stabilize the cell position within the other cells. So kind of forming connection between cell and its neighboring cell. And then it also helps maintain the shape of the cell. So shape maintenance, stabilizing where your organelles are located, and then making sure where your cell is actually located. Now, the largest of the three that we have, which is microtubules. Microtubules are extremely important in terms of cell division, which is something we learned about last time. So remember spindles that are basically going to move your genetic information or your chromosome in the opposite end, pull them apart. That's made up of microtubules, so spindles requiring during the cell division process. It is involved in changing the cell shape. And then one thing that is not listed here, and I do really want you guys to know, is the movement of the cell or motility of the cell. So please add that to your own notes. Have you guys heard of flagella before? Anybody has heard of flagella before? In what is the most common cell we have in humans that have a flagella? Sperm, correct. The long tail of this sperm is basically identified as a flagella, which is a long, again, long structure. And I'm gonna type it in the chat box so you guys can see the actual name. So flagella is basically the tail of the sperm, which gives it its motility. The other word we have, which is similar to flagella, but they're much smaller, they're more hair-like extensions. They're called cilia, correct? So I am typing and it's kind of hard for me to do both, apparently. Hair-like extension needed for movement. So we also have these structures that are involved in terms of moving molecules. You have a lot of cilia, guys, in your air passageway to clean the air from all the dust particles. So you have cilia and flagella, which are both associated with movement and motility, and they are both made up of microtubules. So make a note for yourself. Again, this is typically an exam question. If I say, what is the most important cytoskeleton associated with movement, then your answer should be microtubules. The last organelles that I'm gonna mention, I believe that's the last one, yeah. It's your centrioles. Centrioles are basically these beautifully structured. I love the structure of this. This is so marvelous, even the picture they've taken. It's a barrel-shaped structure, which is made up of basically sets of microtubules. You don't need to know the numbers. And these are where eventually where your spindles eventually form from. So in terms of, again, the cilia and the sperm that we talked about in terms of microtubules, they originally coming from your centrioles that are going to develop their microtubules to form either the long extensions, which is the flagella, or the literally tiny structures that are called cilia, or the hair extensions called cilia. Okay, so that's it for our lecture we were supposed to do last week. I only took us about an hour. Is there any questions about this PowerPoint, guys? Any clarification about this PowerPoint? Before we switch gears to histology. Okay, give me 1 second. I closed my other. So we're just gonna do about 20 minutes of lecture for histology, and then I will take a break afterwards, guys. So bear with me. Same thing that I mentioned here, guys. Please pay special attention to what I'm going to talk about from this point on, which is basically tissue and integumentary system. Both of these are the lectures that you guys are going to be using in lab today. So if you understand the lecture right now, it will help you significantly completing the lab. So there is a connection there. Okay, I guess there is no questions, so we're gonna go ahead and get started. And again, do about 20 minutes of lecture and really Russian? And then we'll go ahead and take a break. Okay, so if you guys go back to the organizational level we talked about, we said the most basic level of organization was our chemical levels, which were atoms and molecules. And we talked about them in terms of carbs, proteins, nucleic acids, and lipids. And then we said the second level of organization was our cell level. And we talked about that. That was our focus again for the lecture we just finished. We looked at the plasma membrane of the cell, the nucleus of the cells, and all of the organelles and their function. So building up based on that, we have the next thing that we have, which is basically the next level, which is the tissue level. And again, tissue is basically a collection of cells that make up your, make up your organ eventually. So I'm gonna skip some of the slides that you guys have. So please do keep in mind that as we go through this, if I say you don't need the information, don't focus on that. So when we look at the tissues, we have 4 major categories of tissues: epithelial, connective, muscle, and nervous tissue. If I simply want to describe these tissues with one simple function in front of them, and it's not the only function, but simply put, you have epithelial are responsible usually for covering, connective tissue is responsible for support, muscle tissues are typically associated with movement, which kind of makes sense. And nervous tissue is associated with control or coordination, since they're associated with your nervous system. Again, it's more complicated than this, but this is kind of like an overview of your tissue. So when you're looking at epithelial tissue, which is the first tissue that we mentioned earlier, these tissues have, if I want to go into a little more detail about their function, protection is a really important function for these. So if you think about your skin, the top layer of your skin, what I'm touching right now, this is basically epithelial tissue. And the goal of having this skin is because you want to protect the underlying tissue that's right underneath it. You also have epithelial tissue that plays role in absorption in things like your kidney and your digestive system. Secretion also is involved in your kidneys and your respiratory system, as well as the ion transport. Filtration, really important in your kidney. And then forming slippery surfaces, which basically can be found in any organs in your body. Forming that slippery surface is done by your epithelial tissue. Now, I want to simplify this for you guys. Whenever you look at your epithelia, so epithelia has two part names. It has a first name and it has a last name. The first name of your cell is associated with how many layers of cells are stacked on top of each other. And again, this, what I'm describing to you guys, cannot be applied to other connective, other tissues like connective, muscle, or nervous tissue. It is specific to your epithelial tissue. So when you're writing an epithelial tissue, you have to think about 2 things. How many layers of the cell do I have? That's first question. And the second question, which forms the second or last name, is what is the shape of my cells? So how many layers and what is the shape of the cell? If I simply have one layer of the cell, I'm gonna use the word simple. If I have more than one layer of cells, doesn't matter how many, guys, as far as it's more than one, it would be referred to as a stratified. So it could be simply 2 layers, or it could be 20 or 30 layers of cells. As far as it's multiple layers, it's called a stratified. Now, what about the shape of your cell? You can have epithelial cells that are called squamous epithelia. Squamous literally means flattened cells. So flattened cells are basically kind of look like squished. Cuboidal cell, as the name implies, it's cube shape. So they're kind of more like a square shape if you look at them. And then columnar cells are going to be long cells or elongated cells, more like literally like columns. And then you have this extra category, which is called pseudo-stratified columnar, which simply put means they appear to be multiple layers of columnar cells on top of each other. But what is the word pseudo implies, guys? When you hear the word pseudo, what does that mean? Anybody? That was what pseudo means? Fake, correct. Thank you. So pseudo-stratified columnar simply means that you are doing it appears that you have multiple layers of long cells stacked on top of each other, but in reality, that's not the case. It's only a single layer of flattened cells. Sorry, it's a single layer of columnar cells. Okay, so let's take a look at this, guys. Look at these cells, guys. So notice how flat these cells look. So like it shows here, they look like squished, right? The nucleus is more oval shape, like extended, like somebody is putting pressure on it. You have cuboidal cells that are again more square shape, and then columnar cells, which are long or elongated. Here is you have one single layer of cell, and then you have many layers of cells stacked on top of each other. So let me, based on what I've told you, and based on the picture you have here, let me ask you a question. So what is, if I say, what is the name of a tissue that's made up of 10 layers of flattened cells? What would you call that epithelial tissue? 10 layers of flattened cells. I need an answer. Okay, so again, many of you guys are just saying stratified. Remember, stratified is just telling me how many layers you are, but there's 2 parts to your name, right? So first name and last name. First name is how many layers, then the shape of the cell. So the full name would be what? Stratified squamous epithelium, right? You cannot just say stratified or simple because that doesn't distinguish what is the shape of the cell. So if I say, let's look at another example. If I say a single layer of a square-shaped cells, what would you say? Single layer of a square-shaped cells. What would you say? Simple cuboidal, correct. So again, a stratified is multiple layers. So simple means one layer, cuboidal means there are square shape, correct. And then what if I say many layers of elongated epithelia? What would you call that? Many layers of elongated cells. A stratified columnar, correct, you got it. So don't make it complicated for yourself. Always remember, guys, that there are 2 parts to the name. Don't just write columnar or stratified. Don't write simple or squamous. It has to come together. The full word is the combination of both of those together. So make sure you guys remember that. So I provided these pictures for you, which is again a really good help in terms of your lab as well as your lab exam that's coming up in a couple of weeks or a few weeks. So right here, you see an example of lung tissue, which is also something that you need to know. example for each tissue. So the lung tissue, the lining that you see right here, this is your lung tissue. And I don't know if you can clearly see that. You can, of course, kind of look at your own PowerPoint and zoom into it. But you see these dark spots through them? Those are the nucleus. So this one, these ones, these are the nucleus. And notice the nucleus is not this beautiful round nucleus. They kind of look flattened. So that means that these cells are flattened and a single layer of cells. So they're simple squamous epithelium. And again, examples of the location, there's a bunch of location listed here, but the best example that you can see also here would have been your lung tissue. Now here is another example. So here, I'm gonna just use my pen to kind of show you. So these that you see, these round things, it's actually made up of multiple of the cuboidal cells. So kind of look like this. This is one cell if you wanna kind of look at it. I'm just kind of highlighting it for you. So this is one cell, here is another cell, kind of giving you the border of these. And a lot of my students, when they see this, they're like, these don't look cube shape. Guys, remember, we don't have a ruler inside our cells that say, oh, it's not a perfect cube, so we're not gonna consider this cuboidal. It's as close as possible to what we can see in real life. That's where these names come from. So notice that each of these are one single structure. It doesn't mean that these are stacked on top of each other, guys. So this is one layer of cells. This is another layer of cells. They're not stacked on top of each other. An example of this would be the kidneys. So kidneys is a really good example of where you can find a lot of simple cuboidal cells. One thing that I tell my students, for the level that you guys have, we don't see a stratified cuboidal, and we don't discuss a stratified columnar, okay? I have pictures of these, but for you guys, those two types of tissues are so uncommon that unless you're taking pathology and looking at tissues in detail, you're not gonna see those. So when you look at the sample, if you think it's a stratified columnar, or if you think about a stratified cuboidal, the chances are they're not. They probably are just simple cuboidal and simple columnar, because they are so much more common than a stratified cuboidal and a stratified columnar. So here's another example of the cell. Right here we have the gap. So this is the gap, which means these are not stacked on top of each other. They're separated. So here is a long cell, right? Here's one cell, here's another one. So these are one layer of cells that are stacked. They're not stacked. They're single layer of cells that are elongated. So that makes them simple columnar epithelium. And correct. And the best example for the simple columnar epithelium would be your digestive system. That could be your stomach, your large intestine, or a small intestine. Any of those would be a good example, as you can see highlighted in the image here. The next we have, I don't know if I actually talked to you guys about this. This is pseudostratified ciliated columnar. So this is an example of the pseudostratified columnar epithelium. So see how these cells look long? So as we're talking about this part, anything above this part. So these dots that you see inside, these are the nucleus. And it seems kind of like you have nucleus on top of the nucleus, but in reality, they're not. They're just kind of stacked like this. And I'm just gonna do a quick drawing for you guys. So you guys can, and this is like a rough drawing. So hopefully you get the idea. So they kind of look like this. So if you're looking at this, see how it seems like this one is a stack on top of the other one. So you think, oh, these are stratified. But in reality, these cells are just kind of smushed together. So the nuclei are so close together that they look like a stack. So the example of this is actually, again, pseudo-stratified columnar epithelium. So not an actual stratified columnar. Again, that one is really rare. We don't see that. We only see it in the urethra sample. So a stratified epithelium, if we're talking about a stratified again, we're talking about more than a one layer cell stacking on top of each other. You have three types, squamous, cuboidal, and columnar, right? Based on the shape of the cells. This one, if you guys looking at this, what do you think this is look like? Are these cube-shaped, long cells, or flattened cells? So stratified squamous, correct. So these are flattened cells. Like you can clearly see that there are many layers of cells on top of each other, right? And they are squeezed, right? They're not this beautiful cube shape or they're elongated cell. So really common, again, as I mentioned, in terms of a stratified squamous epithelium is in your skin. So what you're touching, this layer, what you actually scrub and get rid of, the dead skin cells, what you're getting rid of is the stratified squamous epithelium tissue. These two, again, I provided an example for you, but they are very uncommon in our body. So you can see this one, you have cube-shaped cells, okay? They're stacked on top of each other. This means that two layers means they're stratified. They're cube-shaped, so they're cuboidal. All you need to know for this one, guys, is where they're located. So if I say, give me an example of a stratified cuboidal cell, you will say salivary gland, and that's it, you're done. That's all you need to know. The next picture, you can see the cells are more elongated. Ooh, I don't know what happened. So you can see the cells are more elongated. So here is one cell, here is another cell. So these are columnar cells, again, stacked on top of each other. So that makes it a stratified columnar. And the example is found inside your urethra. And it's more common in actually male urethra because male urethra is longer. In female urethra, usually just stick to the stratified squamous epithelium, but you don't need to know that detail. I don't need you guys to know the transitional epithelium in detail. The only thing that I need you guys to know about transitional epithelium is the location. Literally, all I need you guys to do, highlight this for yourself. If you hear transitional epithelium and where you can find it, the answer would be bladder. I'm gonna give you a brief detail, but it's not something that you really need to know. Transitional epithelium basically means they kind of look chubby when they start, but as they stretch, they become flat in shape. So they're transitioning, like they're changing, right? So when your bladder is empty, basically, you don't need that stretching. It's like a balloon. So the cells are more chubby. As the bladder starts to fill up with urine, basically you're stretching the tissue, right? So your cells that are kind of chubby, they start to stretch and they transition. So they become more flattened and elongated. So that's an example you can see in your bladder. You can see it in the umbilical cord. You can see it in uterus, which is not listed here. But you can see those transitional epithelium present in those tissues. And here is the picture, basically kind of chubby cells when your bladder is relaxed. And then as soon as the bladder starts to fill up with urine, you can see that the cells are kind of flattening up. Okay, what time is it? All right, so let's go ahead and take our break until 9:35. And then we'll continue with this and we'll see how far we get. And we'll go from there. So please be back by 9:35. Hi guys, so we're going to continue with our next tissue, which is going to be, I believe, connective tissue. So we talked about epithelial tissue. Make sure you guys know the 7 types of epithelial tissue and make sure you guys can describe them and also provide an example of where each of these tissues can be found. So let's look at the connective tissue. So connective tissue are important in terms of protection. They help us with our immunity in terms of things like areolar tissue. They're found inside our blood vessels and around our nerves. They are helping us with connecting the cells that are found in our body in terms of forming junctions we talked about last time, like things like tight junction, desmosomes, or gap junctions. They help us store nutrients like your fat tissue that's considered to be a connective tissue. So looking at the overall function, connective tissue is really general. And you guys are going to take a look at this in terms of what those look like. Now, when we look at connective tissue, one of the things that you guys need to remember is that it is, okay, so let me back up. When you talk about epithelial tissue, you can kind of see epithelial tissue, how you have one cell, one next to it, next, and then you can have cells that stack on top of each other. But when we're talking about connective tissue, you don't have that frequent cells. So you have cells here and there. And then you have this kind of a background space that's typically filled with either proteins or some sort of fluid or solid material like calcium. So that background where the space is between the cells, basically, that space is referred to as the extracellular matrix, which sounds familiar since we already talked about it. So we're going to take a look at not only the cells, but how these extracellular matrix actually differ from one cell, or sort of one type of tissue to another type of tissue. So this chart is really helpful in terms of identifying and creating that kind of a flow chart for the connective tissue. So when we look at the connective tissue, there are three major categories in terms of functioning for the connective tissue. We have things that are called proper tissue, connective tissue proper. We have tissues that are fluid, which basically means they're liquid. And then the ones that are acting as supporting tissues. So things like bones or cartilage. So when we look at the proper tissue, they could be loose or dense. And basically what that means in terms of loose or dense is when we look at the space, the background, that extracellular fluid, do they have a lot of protein fibers? If it is, then they're identified as dense. If those proteins are almost very loose or very infrequent or non-existent, then we refer to those as loose connective tissue. So loose connective tissue examples are things like areolar connective tissue, adipose connective tissue, and reticular connective tissue. Dense connective tissue are either dense regular or dense irregular. I don't talk about the elastic with you guys. So dense regular simply means, are these protein fibers moving in one direction? Are these beautifully organized? Then they're regular. If they're kind of chaotic and they're perpendicular to each other, they're all over the place, then they are identified as dense irregular. When we're talking about the fluid connective tissue, which is the middle chart, fluid connective tissue are either your blood. Yes, blood is a connective tissue, which is basically contain your fluid portion like plasma, and then red blood cells, white blood cells, and platelets. And then what we call lymph, which is basically found in your lymphatic vessels. Supportive, basically they're responsible for providing support or a structure for your body. These are either cartilage, which have their own three types, hyaline, elastic, and fibro, or your bone, which is basically the solid bony structure that forms your skeletal system. So let's take a look at each of these connective tissue. Again, we're keeping things really simple, guys. We're trying to go as basic as possible in terms of what you need to know for this tissue and identification. So I'm gonna ignore this slide for you guys. You don't need to know about this. As far as you know it for each of the tissue, you don't need to specifically know this type of information. So loose connective tissue, again, anytime you hear loose connective tissue, think about the fibers are being very low or almost non-existent. And again, 3 types: areolar, adipose, reticular. And then if you have lots of connective tissue, are they regularly organized or are they all over the place and identified as irregular? So areolar connective tissue, keeping it simple, guys. Again, know the picture. This is a common picture I give you for areolar. What you need to know for them: all of these lines that are going through these, so they're thick lines, which are kind of like this, like darker spots, versus these kind of a string-like almost. These are called fibers. These are your protein fibers. Do you need to identify which type of protein fiber? No. All you need to know is these lines that are going through your image are fibers. And then the little dots that you see, so all of these structures, these ones, these are cells that are found in the areolar tissue. Do you need to know what type of cells they are? No. Do you need to know what type of fiber they are? No. All you need to know is, can you identify this image as areolar? Yes or no? Do you know what those lines imply? Fibers. What are those kind of blobs or circles or oval structures look or represent? They represent your cells. Don't care what type of cells. Where they can be found. That's another part that you need to know. So where you can find them, right, done. It's underneath your skin or as part of your skin. Another place that you can find them is connecting your skin to your muscles. As you can see in the picture right here, this is basically a picture that shows inside of the abdominal cavity, connecting the internal organs to the muscles of the abdomen. You don't need to know any of these. So I'm gonna eliminate this for you guys. So again, limit your knowledge. We're just gonna kind of focus on the essentials of the sample. Adipose tissue. As the name adipose implies, hopefully most of you know, or hopefully all of you know, adipose implies fat, right? So adipose tissue is basically going to be storing fat. So when it says nutrients, what I'm talking about specifically is, oops, sorry, it's fat molecules. So adipose basically means fat. What else do you need to know for the adipose tissue? Again, know the image. So these kind of like blobs that kind of look like almost clear lining and nothing inside of it. That's a single cell of adipose tissue, which is called adipocyte. And locations of them. Again, location, you find it under your skin as hypodermis. We already talked about that. You can also find it in your heart, your lymph nodes, around your eyes, and some of your muscles. And most of these is begin. What is the job of having this fat tissue? It's just to store energy. That's the most important job you have for these fat tissues. Reticular tissue. What do you need to know for them? First of all, be able to identify this as reticular tissue. How do you identify this? To me, the best way to describe this is kind of look like grapes, right? When you buy a kind of a bunch of grapes, you can kind of see the vine, right? And then all of these dots are your actual grapes. So if a tissue looks like grapes, that's your reticular connective tissue. And then know the location. Any of these locations will suffice. Your spleen, your lymph node, your thymus, and bone marrow, which basically means most of your lymphatic tissue contain reticular tissue. Simple, guys. Don't go into detail. Don't go down the rabbit hole to know more about this. We're keeping it simple. I don't need you guys to know about the cells. I don't need you guys to know about the fibers. As far as you can identify this as a reticular connective tissue and provide an example for me, you're good to go. That's it. Nothing more than that. Don't make it more complicated. Now, when we're looking at the connective tissue that is dense, which means lots and lots of protein fibers. Look at this picture, guys. You can clearly see a distinction. This is like a very calm sea, right? Very beautiful. All of these lines that you see, they kind of move in one direction. That makes this dense, regular connective tissue. All the protein fibers move in one direction or are parallel to each other. Where can I find this? All you need to know: locations, ligaments, and tendons. Dense, irregular connective tissue. Again, look, chaotic, right? All of these protein fibers are all over the place. There's no organization. There's no beauty to it. Well, there's beauty, but not as beauty as dense, regular. So where is the best example for dense, irregular connective tissue? The dermis of your skin, where you have all of your oil gland, your sweat glands, your hair follicle. That's the dermis of your skin. And that's where the tissue can be found, dense, irregular connective tissue. Is this making sense so far, guys? Is this clear as far as what I need you guys to know for your exam? Any question, anything that just doesn't click for you guys so far? Okay. Please, if something just doesn't make sense, type the question in your chat box and I'll get to it. Okay, so for the fluid portion, we talked about blood, right? So blood is one of the main type of connective tissue, which basically is fluid or it's liquid. Now, in terms of blood, there's a couple things that you guys need to identify. First, if I say, what's the function of blood? Function of blood is to transport nutrients, waste, and gases. Nutrients like kind of like glucose, waste like CO2, and gases like oxygen. Now, what do you need to know for blood? Most important things you need to know is there are three types of cells, clearly. This is three types of cells: red blood cells or erythrocytes, white blood cells or leukocytes, and then thrombocytes or platelets. What is the job of your red blood cell? Red blood cells is responsible for transporting oxygen. Leukocytes or white blood cells are responsible for your defense or immunity. And then platelets are responsible for blood clotting. So if you cut yourself, what stops you from bleeding to death is your platelets. If a person has a low platelet count, that basically means that they might be more susceptible to severe bleedings. Next thing you want to be able to do, next thing you need to know is being able to identify it on an image. So if you're looking at all of these pink circles, and they're not white at the center, it's just because there's a lot of light going through your sample, it looks kind of empty at the center. But in reality, they appear as all pink, these tiny pink circles. These would be your most common type of cells, which is your red blood cell. Hence, your color of your blood is red. Then you have these kind of larger cells, typically 1.5 to twice the size of your red blood cells. And they have very pronounced nucleus, this purple stuff that you see inside of them, that's the nucleus. These are identified as your white blood cells. So you have red blood cells, which are majority of your cells, and then white blood cells, which are these gigantic structures with a very purple nucleus inside. And then you see these little fragments? These are your platelets. Platelets or thrombocytes, which are responsible for basically blood clotting. And again, you don't see as many of them because they're not as frequent. So in terms of numbers, the highest are your red blood cells. You see a few white blood cells. And then kind of scattered between these type of cells, you see fragments of cells, which are your platelets. A common exam question, guys: identify the tissue, identify the cell type, and then give me the function for each cell type. Neutrophils, so when it's identified as lymphocytes or neutrophils, you learn about them for the blood lecture. They're basically classifications of white blood cells. So if you see neutrophils here and lymphocytes here, or eosinophils or basophils, all of these are categories of white blood cells. At this point, for your lecture and for your lab, I don't need you to identify that. All you need to say is white blood cells. That's it. We're keeping it simple. Okay, guys, cartilage. Surprisingly, we actually have 3 different cartilage types. Really cool fact about cartilage, which I think a lot of people miss. Cartilage has no blood vessels and no nerves. So when you think about, like, I always give the example, when you kind of look at your ear, like this portion of your ear, a lot of times it's relatively, and I'm going to say relatively, painless, because the cartilage portion of that tissue is basically doesn't have any nerves, and you also don't bleed if you get an earring in that section, because there is no blood vessels. So lack of blood vessels and nerves are characteristics of the cartilage tissue. Most of the cartilage is actually water. So 80% of your cartilage is water. Doesn't matter what type of cartilage we're talking about. Now doesn't matter again, whatever cartilage type we have, they only have one type of cells inside of it, and they're called chondrocyte. We've seen the word chondro before, if you remember hypochondriac. So chondro means cartilage and cyte means cell. So cartilage cells are chondrocytes. There are 3 types of cartilages, guys: hyaline, elastic, and fibrocartilage. Simple, guys. Don't make it more complicated than what it is. Hyaline cartilage, if you look at these, all of these dots that you see, the white dots that you see in the tissue, with the little purple spots in the middle, those identify your chondrocytes, okay? So the white stuff through, that's your chondrocyte. The background, if you look at the background of this picture, guys, look how clean it is. It's like somebody took a pencil and just gently went into the background with nothing very visible. So you have this really glossy, beautiful, uniform background, and then you have your cells sporadically placed in your tissue. That is the characteristic of hyaline cartilage. Okay, so very clean background, and then your chondrocyte is spread throughout your tissue. So no fibers, no protein fibers are visible in this tissue. Where can you find hyaline cartilage? Any of these examples would do. You have them in your joints, you have them in your trachea, which is the air passageway or your air pipe. You have it in your nose, so the cartilage part of your nose, the movable part, that's hyaline cartilage. And then fetal skeleton. So babies, or fetuses I should say, up to the first 2 months during pregnancy, they actually don't have bones. They have hyaline cartilage that looks like bones, but they're not actually bones, which I think always fascinates me. No bones. Even when they're born, their bones are still pretty soft, so just FYI. Now, elastic cartilage still have those chondrocytes. So see, this is kind of like a zoom-in picture. You have these are your chondrocytes. But what you notice about the background on this image, guys, look, compare this. Really clean, really uniform, right? And then what do you see? It's kind of like somebody do the background and then took another color and just did a darker lines throughout the sample. So that's basically fibers that are going to be a stain when you look at them under the microscope. So you don't have that beautiful glossy looking for elastic cartilage. Again, the best examples for elastic, look at this, guys. E for E, elastic found inside your ear, and epiglottis. Anybody has heard of epiglottis? The word might be familiar to some people. So have you guys heard of the little flap that you guys have on your air passageway that basically prevents food from going down your air and goes down your throat, basically, or your esophagus? And then basically if this flap doesn't work properly, it causes you to choke if you're eating something. That's what epiglottis is. It's this little flap on your air passageway. So when you choke, it means that epiglottis failed to do its job, basically. And the food has gone down your air passageway instead of your food passageway. It's kind of cool. I think it's kind of cool. I'm kind of boring. I geek, I guess, sometimes. Okay, fibrocartilage. What's the distinguishing characteristic about fibrocartilage? Best way to look at fibrocartilage is that it has that really pretty blue-purple color combination to it. So if I give you a fibrocartilage, the way it always stains the blue and purple color. And then the chondrocytes, which are the spots that you see right here, these spots are kind of purplish spots. Like those dots are really beautiful purple, dark purple color. So look for any tissue that is this beautifully wavy blue purplish color, and that's your fibrocartilage. No other tissue would look like this with this color under the microscope. Where can you find your fibrocartilage? Best place is your intervertebral disc. So you know how like people say, oh, I have a disc problem. Basically, they have back problem, or they call them, they have disc problem. They have to do surgeries, typically. The main problem is with this type of cartilage, basically. So if you have a vertebra, then you have another vertebra. The gap between them, these bones are not actually in contact with each other. There's a layer of cartilage between them. And again, in many cases, if you damage this cartilage because of trauma or car accidents, typically, then it's really, really difficult to heal. A lot of people actually need surgery if there's damage to this tissue, and they can never recover from it fully. Anybody knows what pubic symphysis is? Anybody? No? Okay. So think about your two hip bones. So here's a hip bone, here's a hip bone. The part in front where basically the genitalia is located, where the two pubic bones come together, how are they held together? They're two separate bones from your left leg and your right leg. So the place where these two bones are actually coming together, there's a tiny cartilage that joins these two pelvic bones together. And that cartilage is a fibrocartilage called pubic symphysis. So if you look at this, if this is the pelvic area, the area in front, right where the genitalia is, there is a layer of cartilage right there. And that's called the pubic symphysis. And unfortunately, and I just always like to add this because you can see this in the skeletal system, pubic symphysis for male and female is supposed to be pretty strong. It's known for its strength. So it says high tensile strength. So it shouldn't basically stretch or be damaged. But during pregnancies, so if any of my ladies are having pregnant, you may actually feel pain in that midsection close to childbirth, because that can stretch sometimes using the hormones to open up the birth canal, so the baby has more room passing through it. It never happens in men though, but in females, if you have given birth, hormones can have an effect on pubic symphysis and loosen it up a little bit to open up the birth canal. So go ladies and pregnancies, yay. I know, it's kind of cool. I think it's fascinating. Among many other changes that women undergo during pregnancy. So yay. Okay, the next tissue we're gonna take a look at is your bone tissue. So bone tissue is, again, the last type of connective tissue we have. The tissue that I'm showing you here is a typical bone that you can see in your arms, your legs, your skull, doesn't matter. This type of bone have these circular units, and I'm just gonna highlight this because, again, this is really important for both your lecture and lab exam. So these circular units that you see for your bone are called osteons. I'm gonna type it in the chat box because it's easier for me than handwriting it. So they're called osteon. So these circular units are called osteons. And then basically what you have is you have these darker spots throughout them. So I'm going to just mark them kind of like these. These are called osteocytes, which, as I say here, osteocytes are basically responsible for bone maintenance. These are your bone cells. So osteocyte, I can't spell. Oh my God, I can't spell. Sorry, guys. It's just simply bone cells, as you can see. You won't see these type of osteoblasts and osteoclasts. So I don't need you guys to know them for now. We'll talk about them later in the skeletal system. And then the other thing that you see is this circle right at the center of your bone, right here. You can see again, one right here and one right here. This is called central canal. And this is where you have your blood vessels and nerves. And then one last thing before I forget, osteocytes that we have, which are these, again, darker spots, are inside a housing called lacuna. So if you're looking at this picture, guys, and make sure you guys are looking at what I'm drawing. So these lines that I've drawn, these lines, see how it says osteocytes inside the lacuna? So in an exam, if I ask you what are these things represent, you can either refer to them as the cavity, which is the lacuna, or the cell, which is osteocyte. Either answer is correct. However, if I ask you what is the cell that is in this space, then your answer would be osteocyte. Because lacuna is not a cell, it's the holding, it's the cavity that holds the cell. So please, 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 and this is like the biggest mistake I see my students do, read the question carefully because you don't know what I'm asking you. So if I say, what is this structure represents? You can say osteocyte or lacuna. But if I specifically say what type of cell, then you have to say osteocyte. Have to be particular. Any questions about the connective tissue, guys? Oh, good, we're doing a good time. No, we're good? Okay. So muscles, again, something about muscles. Osteocytes is, sorry, let me go back. So osteocytes are these little darker spots, like these ones, that kind of go around. Okay, awesome. All right, so let's go ahead and move on to our muscle tissue. So there's three types of muscles that we have. We have a smooth muscle, cardiac muscle, and a skeletal muscle. And before we even move on forward to this, just think about the name, guys. A skeletal muscle, it means muscles that are connected to your what? Use your logic. So when you say a skeletal muscle, these are muscles that are connected to bones, correct? Your skeletal system, correct. Can you guys give me a name of a muscle that you know that's connected to a bone? If you ever worked out, then you should be able to give me at least one name of the muscle. Tricep, deltoid, your quads, correct. So great, biceps, right? Hamstrings, these are your common muscles. Your six packs, right? Your belly muscles, right? Your trapezius, your traps. So that's a skeletal muscle. So use your common sense, guys. Your glutes, right? These are muscles that are connected to your bones. So any of these examples, any of these examples will do. If you're thinking about the word cardiac, what is the word cardiac implies to you? Heart. So the only place, if you logically think about this, the only place you're gonna find your cardiac muscle would be your heart, correct? So a smooth muscle, which is the last category of muscles we have, is anywhere else in a body where I can find something that resembles a muscle, but it's not cardiac muscle or skeletal muscle. So that muscle can be found in your intestines, in your stomach, because they move, right? Your intestines do move to move the food. Your stomach definitely move, otherwise you will never throw up, right? Or digest food. The iris of your eye also have a smooth muscle. Uterus is a really good example of this smooth muscle. It's a really thick layer of a smooth muscle, correct? Because it gives it contraction to be able to push the baby out. So when you're looking at these, just by looking at the name, guys, start with that. You can distinguish where each of these muscles can be found. Now, when we look at the muscle tissue, we're looking at characteristics of the muscles. So one of the characteristics is presence of what we call striations. So when you hear the word striation, think about stripes, like a zebra. So if your muscle is going this way, so imagine a muscle that's this way, your striations are going to be going up and down, up and down, up and down, right? So cell is like this way, and the stripes are going this way. Okay, so they're not parallel to the muscle. Intercalated disc is another thing. And so intercalated disc is the connection point between one cell and another cell. If you guys remember desmosomes from our previous lecture, desmosomes are found inside the heart to help you spread the tension. So if your heart muscle is continuously pumping, you don't want that muscles to basically rip apart because there's a lot of tension in that muscle. You want to evenly distribute the tension in your muscle. And the way you do that is by having these intercalated discs that strengthen the tissue and spread the tension. Now, the only muscle in our body that continuously works, right, that never gets a break, is not your skeletal muscle or a smooth muscle. It's your cardiac muscle. That's the one that's going to have the intercalated disc because it's continuously under tension. So just make sense of this for yourself. Don't make it more difficult or more challenging. Intercalated disc is associated with tension. The only muscle that has a lot of tension is your heart because it's beat all the time. So it can only be found in that tissue. So let's take a look at the characteristics that distinguish these muscles. So if I look at an individual, one single cell of the smooth muscle, it's a spindle shape, which means it's kind of chubby at the center and narrows down at the end. Now, when I say this to my students go like, well, I can see this in a tissue. That is true. In your tissue, you can't see it because you're looking at hundreds upon hundreds of cells stacked right next to each other, and you won't be able to see one single cell. What your tissue would more look like is something like this, kind of this beautiful uniform cells all moving in one direction. Okay, so you can see this image. Don't ever draw this image for me when I ask a smooth muscle tissue, because that's single cell, not a tissue. But what you would draw is kind of like this beautiful pattern, a uniform pattern of cells that all are beautifully stacked next to each other. The other characteristic a smooth muscle has is each of the cells. So if I count each of the cells, they have this beautiful nucleus inside of them, but it's a single nucleus, like every common cell that we have. So one nucleus per cell, which is basically what you see as these dark spots throughout your image, or like this dark spot right at the center. These muscles have no striations, guys. Now, common misconception. My students say, well, I see light and dark patterns, right? So see, here's the light, here's the dark, here's the light, here's the dark, right? That's the common thing that I hear. Remember this, guys. A striation is if the cell is moving this way, the striation patterns is vertical. So if I'm looking at the cell and I know the direction of my cell is this way, if I want to see a striation pattern should be visible this way. Okay, not horizontally, but vertically. So a smooth muscle, you cannot see that because there is no striation pattern. There is no intercalated disc because we already established that intercalated disc is only cardiac muscle. And again, can I control these muscles? Think about this. Can I control when my iris closes and opens? Can I decide when my stomach decides to digest my food, right? Or when my intestines try to move the food? When my contraction of the uterus starts? None of these are under my control, right? So the movement is identified as involuntary movement, right? Not under my control. Cardiac muscle, what you can see is the striation pattern. So look, here is the muscle going this way, okay? Look at this pattern. So do you see these lights going this way and that way? Okay, those are striation. Muscles in one way, the striations in the opposite direction. You have intercalated disc. Do you see these darker lines right here? Right here, right here. These are all intercalated discs, which means that's the boundary for one cell. Can I control these muscles, guys? Can I control my heart, say when it beats hard, fast, slow? No. So the movement is going to be involuntary, correct? The nucleus, these cells are usually have one nucleus. So one cell, one nucleus, one dark spot inside it. However, because these cells can get kind of big, they can have up to 2 nuclei. So some of the cells have 2 nuclei. The only thing that is missing from the information here is the shape of the cardiac muscle cells. So cardiac muscle cell shape, the way we describe it, is branched. So what I mean by branched is, look, if this is one cell, look, it starts from here, and then there's a branch up there, okay? So the extension of the cell is 1, right? And then it forms 2 branches, and it moves on forward. So you have this kind of a branching and opening that's a characteristic of the cardiac muscle. So a smooth muscle was a spindle shape, and then a striation, the shape of the cardiac muscle would be branched as then it starts to form branch. These lines that you see, striations are the pattern you see here. So you can see kind of the stripe patterns going throughout vertically, going up and down. It's not a uniform color. You can see dark and light patterns going through it. That's what a striation is. And again, the location of this is your heart, right? Cardiac, the only place you can find it is your heart. So last one, branch simply means that, so this, like a branch of a tree. So it's like, what is a branch? So if this is 1, see how it's branching to 2, and then it continues and it forms these branches down here. So this is branching. So it's not one cell and you're like, oh, I'm done, right? They're not that beautifully next to each other. You actually form these kind of like branches of trees structure. Okay, a skeletal muscle. So a skeletal muscle is the one that basically, again, any muscle that's connected to a bone. The shape of these cells are cylindrical and long. And I mean by long, between 1 inch up to 12 inches, guys. That's pretty long. So we have one cell, for instance, one single cell, guys, that can extend from the tip of your shoulder to your elbow. That's one single cell. So these cells can be pretty long. Because they're very long, they can have multiple nuclei inside of them. So they are multinucleated. You cannot find a skeletal muscle that is not multinucleated. They have to have multiple nuclei to be able to manage the cell, basically. So a striation, if you guys look at this picture, guys, again, highlight this. This is really important, guys. So in this image on the top, my muscle is going vertically. The image they just took is the muscles going vertically. The striation, do you see the pattern right here? It's a dark purple, light purple. That's the striation pattern we're talking about. So that striation pattern is present in the skeletal muscle as well. And again, the movement is voluntary movement. We can control where that muscles goes. So yes, striation is present both in the skeletal muscle and the cardiac muscle. And a smooth muscle lacks that characteristics. Any question before we move on to our last tissue, guys? I think that makes sense. Awesome. So again, this is a really good practice, guys. When you guys do your lab today, you guys get some more practice on this as you go through the lab. So nervous tissue, pretty basic. I'm not gonna go into detail. This is literally all you need to know. So nervous tissue can be found in your brain, your spinal cord, or basically the nerves of your body. There are two main types of nerve cells that you have, or cells that you have, I guess I should say. Neurons, which is this really large structure you see right here. What the best way to describe neurons are, they have three parts to them. This really long extension. So this extension that's by itself, that's an axon. You have this chubby center. That's the cell body or soma. And then these extensions, the smaller extensions that's coming off of it, they kind of look like an alien. They're called dendrites. So make sure for the exam, guys, if I ask you what is the cell, then you say neuron. If I ask you identify each of the structures within that cell, then your answer is either soma, dendrite, or axon. And then lastly, for your nervous tissue, you have these tiny cells that you see right here around the cell. These are called supporting cells or neuroglia. And basically, as the word supporting cells imply, they help with supporting your neurons. So your neurons are extremely important because they're the ones that actually do the job. But without the supporting staff, then they're basically nothing. Then supporting staff are the ones that are basically nurture them, they insulate them, and they protect them from the environment. Very funny. Okay, any question about this PowerPoint as a whole, guys? Here's another picture of the nervous tissue. No? That's awesome. I'm glad. It's fascinating when you understand. I think a lot of times my problem is when there is things that are in the news, they don't explain the background. So taking a class like this is generally helpful to kind of understand where the science comes from. So I think it does help a lot. Yeah. So you might have a basic understanding of what they're saying, but when you have the background knowledge, then you can explain or kind of question why the thing they're saying is correct or incorrect. That's why I like especially for now, which if you were in a physiology class, I would have spent a lot of time on, would be lymphatic system and immune system, because of this whole COVID, and there's a lot of misinformation, going out about the virus, about the vaccination, etc. So it would have been a really good opportunity, but we don't have time for that. Sorry. I know immune system is rocks. Although I had a teacher that would talk for like 3 hours, just the most monotone person I've ever had. This guy probably taught for like 40 plus years, and he had like the most monotone voice for 3 hours. He would lecture, and it was just exhausting to listen to him. CD4, CD5, CD8. I don't teach physiology, sorry. I don't teach physiology at your campus. On my main campus, I do. Oh, that's funny. Sorry. I'm just reading your comments. I teach anatomy and physiology. That's my major course, but I only teach anatomy at your campus. Honestly, don't tell that to anybody. But I'm just too lazy to make, because if I want to teach physiology at your campus, which is my favorite subject of all time, I have to do it the way they want me to teach it, and I don't like that. So yeah, my main campus is San Bernardino Valley. So nothing near LA. Yeah, so I'm a commuter. I live in Fullerton. I think I mentioned this to you guys. I live in Fullerton and I commute to San Bernardino, Inland Empire, and then Los Angeles. So no. It honestly, with one on the way and a 2-year-old at home, I barely have time to grade your things. But you're welcome to bring your questions to me. I'd be happy to answer them. But would I teach it? No. I like literally, I'm barely teaching one class and surviving. You guys, if you guys come to my office hours, you can hear my little one screaming and yelling while my husband's trying to get an hour of sleep from his night shift, so he can take her. So otherwise there would be a lot of background right now. Well, you guys don't care, but there is direction. So, but honestly, I think if you guys, and I really mean this, it doesn't matter who teaches the class, because I'm sure there are wonderful instructors at LA, because otherwise they wouldn't have the job. They've been there for a long time. But if you guys, my best suggestion for you guys, if you want to do well in physiology, make sure you remember anatomy, because a lot of people think that, oh, I'm done with anatomy, I'm good, I'm gonna go take physiology. But if you're going into nursing programs, understanding your anatomy and physiology is going to help you significantly in the first year of, or first semester of nursing, because you're going to be focusing on fundamentals of nursing, basically, which is what we're teaching here, like circulation in the heart, you know, how your lungs work, the pressures of the lungs. And these are the numbers you're going to be working with as a nurse, right? Ion concentration, like sodium, potassium, etc. So things like this, learn it now, learn it well in both anatomy and physiology, because it makes your job a lot easier when you go into the nursing program. That's my best recommendation for you. So, sad face. All right, guys. I know we've gone through a lot of information, but I believe your exam is coming up next week, right? If I'm not wrong. And we do need to finish this last lecture, which is integumentary system. But the good thing is that when you guys do this, again, you guys gonna get exposure in the lab on it too. So it's a good way for you guys to review it. Okay, so everybody sees my screen. I'm gonna do the last lecture for today. I know, bear with me, guys. Exam, as I mentioned earlier, and you guys can find that information also in your thing. When you go to your quiz list, it tells you exam when it's open, but it happens at the like 2 minutes before the start of your class next week, and it's open until I believe 9:40. But the time you have to take the exam is 1 hour 20 minutes. I have to look back because I don't remember how many questions I gave you guys. The exams are open. Note, you guys are welcome to take the exam anytime using any of your notes. It's not like I'm gonna come. We don't do proctoria, even though some instructors do. Honestly, I am telling you this, and I told you this on the first day. Don't assume just because it's open notes you're gonna do well in the exam, because when the students focus on the notes, they lose the chance to complete the exam. So I have a student that did really well the first half of the exam, and then the second half of the exam is literally all unanswered questions, because they were so focused to find the answers to the first half, and I'm not giving you that much time. You either know the answer or you don't know the answer for most of the questions. So leave looking at your notes for the questions that you are really challenged, rather than relying it for every question in the exam. I can show you guys where the quiz is after, but we are on the time, so I am going to talk. Okay, so here's what we're gonna do. I'm gonna start the integumentary lecture, leave your questions for now, and then when you guys come back from your break to start your lab, then I'll go over some of the questions that you guys just posted. But I do want to finish the integumentary system by 11:10. So you guys don't have to go over the time for the lecture. Well, all of the questions that you have posted, I will answer them after you guys come back from your break. And I have broken down you guys to your breakdown sessions, okay? But for now, let's just go ahead and start our lecture. Keep your questions or type them and I can come back to them and answer them later. Okay, so integumentary system is our first system that we're gonna talk about today, or for the semester, I guess I should say. If you guys remember the organizational level, we always go back to that. So we did the chemical level, we did the cell level, we did the tissue level. We're gonna skip the organ portion or organ level of organization and just jump into the organ systems. And when we talk about the organ system, when we talk about all the organs within that organ system as well. So the first system that we're talking about here is basically the integumentary system, which is one of the most basic systems we have in our body. The job of this is protection. So basically, when you look at your skin, if it's intact, it's protecting you from your external environment by basically protecting you against viruses, pathogen, etc. It helps you regulate your body temperature. And this is the one that sometimes the students have questions about. So think about your body temperature as when you're hot, you look flushed, right? So your skin tone changes basically. The main reason for that is your blood vessels that are found in your skin dilate or open up, and that causes the heat to literally, the extra heat to radiate out of your skin. So that's how you control when your body temperature is high. So you want to return it back to normal. The other end is when you're in a cold environment, your skin look paler than normal. And the reason for it is because you have what we call vasoconstriction. So your blood vessels constrict or tightens, and that prevents the heat loss from your skin. So you're preventing heat loss by making sure that the heat doesn't go to the surface of your body or to your skin. Cutaneous sensation is basically means your sense of touch, and then excretion of waste. So when you think about the waste product that we have, we're talking about your sweat, you're releasing a lot of your extra ions, as well as ammonia to the surface of your skin. And that's basically kind of acts very similar to your urinary system by removing some of the waste. Urinary system just does it much more effectively. What included in your integumentary system is, of course, your skin. And within your skin, you have your hair, your nails, sweat glands, and what we call sebaceous or oil glands. So for some of these, I provide a little bit more detail compared to the other components. But we are going to start with the skin, which is the main component. So a lot of these words should sound really familiar to you guys, because I think a lot of times we use them in our conversations, but now we're going to put in kind of making sure that we use the correct terminology. So epidermis is the layer that basically, as I said, it's the layer that sits on top of the dermis. So epi means on top of, right? So epidermis is the most superficial aspect of your skin, the one that comes into contact with the external environment. And this is really important, guys. It is the layer that is avascular. Avascular means what, guys? Anybody? Anybody knows what avascular means? No? No, not A1. No vessels. Anytime you have the letter A in front of something, it literally means no. So lack of. So avascular means no blood vessels. So you know how like sometimes when you cut yourself, like you get a paper cut, let's say, and you only see the skin, but there's no bleeding? That means that you only have cut through the epidermal layer. If you do see blood after the cut, that means you have reached the dermis layer, because dermis is when you have the blood vessels. So If it's really superficial cut, you won't see blood because that part doesn't contain any blood vessels. If you do see blood, it means you have cut deeper than the epidermis and you have gone to the dermal layer. So dermis is mostly fibrous connective tissue, which basically means that if you guys recall, I said it's, if you want to write this down or make a note for yourself, is dense, irregular connective tissue. That's what specifically that connective tissue is. And then I wrote hypodermis here, but as you note that hypodermis is technically not considered part of your skin. It's underneath the skin and kind of helps you anchor your skin to the muscles. It is mostly fat tissue, so it helps with both absorption of shock. So literally it helps you, if you hit yourself somewhere, it helps you protect some of that underlying tissue. So the damage to the underlying tissue might not be as severe. And then insulation, we talked about insulation before. Insulation is, it basically helps you keep your core temperature the same. So it prevents the heat loss from your skin. And again, hypodermis or superficial fascia is mostly fat tissue. So you know how when you get an injection in your arms, the layer they're actually doing the injection is in the hypodermis layer. Just FYI if you guys were interested. Here's a picture of your skin. Again, when you look at your skin, the top layer of your skin is epidermis, which is relatively thin. And then the bigger layer underneath, which is where you have your oil gland, your sweat gland, your hair follicle, all of your tactile, your sensations, your receptors, which are like kind of like Merkel cells, etc. These are all located inside the dermis, which is from up here to here. And then you have these yellow blobs at the bottom. And again, this is technically not part of your skin, but it's called hypodermis. And again, hypo means below, right? Below dermis. So the names literally, you have dermis, you have a layer above it, you have a layer below it. So let's take a look at some of the characteristics of your epidermis or your epidermal layer. First of all, what is it made up of? So the type of cells your epidermis made up of is mostly a stratified, a squamous epithelium. Tell me what that means, guys. What is a stratified, a squamous implies? I'm waiting. Multiple layers of flattened cells, correct. So again, not just multi-layer, you have to say a squamous means flat cell. So a stratified means multiple layer, a squamous means flattened cell. I just noticed that I got most of my voice back. Sorry, completely off topic. Keratinized, the word keratinized implied that you have a protein fiber called keratin in it. Keratin is really important because it gives waterproofing characteristic to your skin. So basically, water cannot get in and water cannot be removed from your skin. So the waterproofing characteristic is really important for the keratin, and of course, protection. They're tightly connected with desmosomes. Again, this word, you're gonna see it again and again, guys. Desmosomes are the type of junctions that are going to basically make sure that the tension on your skin is spread evenly. So you don't have, just because you have a point of tension, let's say in your wrist, because there's bones underneath it, that part of the skin is gonna rip apart. So you're gonna have desmosomes, so the tension is evenly distributed within your tissue. And really cool fact, these epidermis layers, these keratinocytes that makes your epidermis, you lose literally millions of them every day. So the top layer, the layer that I'm touching right now, which is called a stratum corneum, and I'll talk about that in just a second. This is literally cells that are dead, and as you move your hand even on top of it, when you go and wash your hands, when you take a bath, when you do, not moisturizing, what is the other one? Oh my God, my brain is not working. What is the thing called when you clean your skin? Exfoliate. Woo! Brain. Okay, when you exfoliate, what you're actually getting rid of, you're cleansing your skin, right? You're removing all of those dead skins from the epidermal layer. What other cells do you have in your epidermis? So majority of the cells you have are your keratinocytes, like 99% of the cells are keratinocytes. However, you have other type of cells. You have cells called melanocytes. So look at the word melanocytes, they produce what we call melanin. So melanin is a protein. A lot of you may have heard of this. Do you guys know what does the job of melanin in your body? Color. So the color of the skin that you see is associated with melanin, among other color pigments, but melanin is the most important one. So basically what you see in difference between somebody that has a fair skin versus darker skin is the amount of melanin they have in their skin, okay? Now, here is a really important fact that you guys need to know. So melanin is normally responsible for, other than the color, which is one of the main functions, is to protect the keratinocyte DNA. So if you have, again, 90+% of your epidermis is keratinocytes, each of these cells have a nucleus, and each of these cells are going to have DNA inside of them. And we established that DNA cannot be damaged, because if it's damaged, it's a good chance it's going to lead to cancer. Okay? So the question is, how do I protect the DNA from one of the most damaging things to the nucleus, which is the UV rays of the sun? So how do I protect? I'm going to repeat this. How can your body protect keratinocytes against UV damage that can change the DNA and cause cancer? And it does that by the help of melanocytes. So melanocytes are very infrequent compared to keratinocytes. But what they do is they produce melanin, pack melanin in these vesicles, and send them to where? To your keratinocytes. So melanin, when they get into the keratinocytes, they go over your DNA in the keratinocytes and kind of form this little cute umbrella over the keratinocytes DNA. Why is that important? Because that melanin now can protect the nucleus from UV damage. It's pretty awesome, right? So I always tell my students, is it a good idea to go to the beach or to tanning booth and change your skin tone or get a tan? The answer is no. Correct. Because what your body is doing is what? It's saying, you're putting me in danger, you're putting me in danger, you're putting me in danger, right? So I'm going to produce more melanin to protect you against that UV, but it can do it up to a certain time. So if you keep having exposure to the UV rays because you keep going to the tanning booth or you keep going to sunbathing, all of these in long term can cause significant damage to skin. People who are who known to have fair skin tones, they're more susceptible to skin cancers like basal cell carcinoma. And basically you're at a high risk of damaging your skin and having more leathery skin at the later age. So don't do it. Lack of vitamin D. So it says, what about lack of vitamin D? So vitamin D is, you do need UV rays. So let me clarify. You do need UV rays with small amounts every day to convert cholesterol to vitamin D. And then yes, you're correct. What we're talking about is sitting hours and hours in the sun with no protection and waiting for that color change to happen. That's what we're talking about. But yes, even when the kids are, when babies are born, when they're dealing with jaundice, it's strongly recommended for the baby to be in the sun to help it produce vitamin D and help the jaundice to go away. So yes and no. It's just the amount. Moderation is the key, I guess I should say. Yes. So that's what I'm saying. So moderation is the key. So it depends on the sun. So there are days and times. What I usually do if I want to go outside is I look at the UV scale. So if you go to your weather channel on your phone, you always have the scale for both the quality of the air and then the UV. So you want to look at the UV and you want to go out when the UVs are at the safe range. So you don't want to go 2 o'clock in the afternoon outside and walk in the sun and sit in the sun for 15 minutes, right? So as far as the UV are in the good range, then your exposure, 15 minutes exposure should be a good, healthy exposure to help you produce vitamin D. And to tell you the truth, sad thing about this is even a lot of people that live in Southern California, they are vitamin D deficient still. So, because they put a lot of sunblock on, and that is a whole different story that can cause cancer, and I'm not gonna get into that. So that's why you take physiology though, not anatomy. Okay, another type of cells that you guys have is called dendritic or Langerhans cells. These cells are very infrequent, but they do provide a role in immunity. So if you cut yourself, these are basically the first line of defense against anything that gets into your body. And what they do is basically they call up the other cells to come in and help them clean up the infection. And then tactile cells, which are also known as Merkel cells. These cells are not necessarily present in the epidermis. They're present at the bottom of the epidermis when the dermal layer starts to form. And they're important in terms of creating your sensation of touch, basically. Okay. So very briefly, when we look at the epidermis has 5 layers itself. And I'm going to keep it simple for you guys. These 5 layers of skin are in order from the bottom going to the top. So from the bottom as in closest to the dermis layer, all the way to the top where you actually come into contact with the outside. You have a stratum basal, spinosum, granulosum, lucidum, and then corneum, which is the bottom layer. The only note that you need to make for this slide, other than the order of them, is that the stratum lucidum is only present in what we call the thick skin, which is things like the bottom of your hands and your feet. So things that are like in a possible risk of being penetrated because of your external environment, they have that extra layer or additional protection in a form of a stratum lucidum. So I'm gonna keep it basic, guys. Don't need to know a lot of information for each of this letter. Basal is the layer, yeah, kind of think about it as the base, correct. It is the deepest layer. And I'll show you in a picture in just a second as well. So a stratum basal or basal layer, by the way, if you don't know what the word stratum means layer. So basal layer, basically. So this is the deepest layer. If I say, why is this so important? Why do I care about this layer? This is what you should come, this is your answer. Because that's where you have mitosis taking place to replace the old cells that you're removing continuously. So if you don't do this, if this layer is not very effective, then you basically run out of epidermis eventually. So these are responsible for mitosis, replacing the old and dead cells that we have on the top layers. A stratum spinosum is called prickly layer. You don't necessarily need to know the second name. I just included it to kind of give you an idea. It's several layer thick, and the most important thing it has that you need to know is that it has desmosomes in it, which means it's dealing with a lot of tensile strength, or it gives it a lot of tensile strength, as in your tissue is not going to rip apart. Granulosum, a stratum granulosum is when you actually start to add keratin into your cells. So the keratinocytes are actually becoming keratinocytes. And this is important, as I mentioned earlier. If you go back to the few slides back, I mentioned that when you start to form keratin, it is important for waterproofing your skin. And that's exactly what is happening here. So water resistance is happening at the stratum granulosum layer. That's all you need to know. Third layer is granulosum. Keratin is added, making your skin water resistant. A stratum lucidum. It's found only in the thick skin, as I mentioned, like the hands and your feet. And it has two or more layers of clear cells. So lucidum literally means lucid, like clear. The thickest layer of your skin that you have in epidermis is a stratum corneum. So corneum basically means horny layer. So this layer is all dead skin cells, right? Dead skin cells. There are about 20 to 30 layers of these stacked on top of each other. And this is the one that comes into contact with your outside. Because it has so much keratin inside of it, extremely important for water, I guess it's extremely important to prevent water loss. And it's a good barrier, right? It protects you again from outside environment, so nothing can basically bring in anything harmful into your body. So here is the layers. This is not a thick skin because we're missing the stratum lucidum. But here, a stratum corneum, granulosum, spinosum, and basal. If I was showing you a thick skin, then in between right here, you would have your stratum lucidum as the fifth layer. And again, notice most of your skin is keratinocytes. There's barely any of the other type of cells present here. Any question, anything that just didn't make sense so far, guys? Good, okay. So dermis layer is the next layer we have. Dermis layer, as we showed in the picture earlier, it has a lot of your components that we associate with the skin, which is the sweat glands, the oil glands, the muscles that makes your hair stand straight, your hair follicle, lot of nerves, lot of blood vessels, and you can see that. Now, when I look at the dermis has two layers. Papillary layer, which is about 20%, and then a reticular layer, which is much thicker, which is underneath the papillary layer. Most of the structures are in reticular layer. So papillary and reticular. Here in this picture, here is your epidermis. Right here is your papillary layer, and then all of this underneath it would be your reticular layer. Why do I care about papillary layer? A couple of things that you need to know about papillary layer is that it does have a lot of protein fibers. I don't need you guys to name them. Just know that it has a lot of protein fibers. What it does contain, it contains something called friction ridges. So friction ridges are basically the fingerprint that you see on your hands. They help you enhance your gripping ability. So kind of like a Spider-Man, it contributes to your sense of touch. And again, every individual has a very particular papillary layer, giving them a very specific fingerprint. So fingerprints is basically your friction ridges. Now, reticular layer, which is the much thicker layer of the dermis, again, it has a lot of protein fibers, but what you need to know associated with reticular layer is these two, what we call cleavage or tension lines and flexure lines. So flexure lines is basically the lines where the skin is connected to the bone underneath it. So skin is connected to the bone underneath it. So you know how when they said they read the palm of your hand, right? The palm of your hand, all the lines that you see at the palm of your hands, the little separation for each of your fingers, okay? What these lines represents, these are flexure lines. So lines that you see at the wrist, line that you see at the bottom of your feet, at your ankles, these lines are called flexure lines. Very visible lines. So like here, again, these are your flexure lines that you see. The cleavage lines or tension lines are not actually visible line, guys. Please make sure they understand this. So don't say, oh, I don't see all of these patterns that they show here. So what you see here, these are cleavage lines that you have in your body. What they represent is the direction of protein fibers that are underneath in your dermis. So protein fibers are, this is protein fibers that are underneath inside your dermis. This is friction ridges are your fingerprints, which are your papillary layer. We're talking about reticular layer right now. So cleavage lines are not visible, guys, but if you look at the protein fibers that are within the dermal layer, within the reticular layer, you can see the following patterns in them. So like in your abdominal cavity, you kind of have these patterns of going oblique. You have these lines that are going horizontal, etc., etc. Why is this matter? So imagine you're a surgeon, okay, and you want to create a cut to do a surgery. The best solution, if possible, I'm not saying that's always the case, which is not usually, the best way to cut is to cut parallel with these lines. When you cut parallel, then it would damage less of these protein fibers and lower the chance of basically significant damage and faster healing, correct? So it does avoid the scarring to some level, correct? Does this always happen? Are you always gonna go with the cleavage line again? The answer is, of course not. A lot of times it depends on, sometimes refer to the preference of the doctor, the location of the surgery, and sometimes you just can't accommodate that. But if the doctor can, they try to go with the pattern of the protein fibers rather than against it to improve the healing chance of that individual or speed up the healing. This is something a lot of people are familiar with, stretch marks and blisters, common terminology that we use. So what is the difference? So a stretching of the skin in a very short period of time, but you basically don't have time to stretch the skin by adding new cells to it. What happens is you start to pull the protein fibers apart. And when those protein fibers are pulled apart, then you get what we call stretch marks. The blisters, on the other hand, is when you have your skin being rubbed against another surface. So because of a friction, what happens is your dermis and epidermis separate from each other. And that the space that is separated gets filled with fluid. And that space, that's what creates the blister. So it's literally a fluid-filled pockets that are separated between epidermis and the dermis layer. Okay. In terms of a stretch mark, there is a lot of, I guess, creams that are on the market right now that do help with removing or lowering the stretch marks. Typically, they don't always go away fully. There are also laser treatments that help enhance the, enhance the regrowth of the protein fibers that were damaged, which also again lowers the scarring or the stretch marks that are present. But I mean, it depends on the person and their body and their genetic to remove and heal the stretch marks too. I know people that were pregnant and, you know, they have severe stretch marks that never went away after their pregnancy, and there are women that, you know, their stretch marks are pretty normal and they go away after their pregnancy. So it literally also depends on the way you take care of it. So the environmental component as well as your genetic. Some people do have a stretch mark, some people just don't. Sorry for those who do. Can't do anything about it. Accept your body. That's the best way I can think of it. Okay. So that's all you need to know for the terms of the skin. So we talked about that within your dermal layer, the thick layer of your skin, you have things like oil glands, sweat glands, hair follicles, and your blood vessels. So let's think about each of these separately. So for hair, what do you need to know for hair? So hair is basically there to protect you against physical trauma. It may not be as well as, let's say, the fat layers in your skin, but it does provide protection. It helps with prevention of heat loss, which kind of makes sense. The hairier the person is, the less heat they're gonna lose to their environment. And shield the skin from sunlight. So again, we learned that sunlight is not good for us. So an additional layer of protection would basically be the hair that covers the body. It also like protects, like if you think about your eyelashes are actually protecting your eyes against UV rays too. And it's kind of like little caps for your eyes. I thought that was kind of cool when I learned about that. Like, it's weird. Okay, another word for hair is pili. So if I ask you, what is actually hair made up of? Hair made up of keratinized dead cells. So it's very similar structurally to what we have for your skin. The difference is that it is more flexible. It does contain more color pigments. So that's where you get the different colors and tones in your hair. And the hair matrix, anytime you guys hear the word matrix, no, it's not the movie Matrix. You're not choosing between the two pills. But hair matrix is basically where your hair is going to grow from. So that's where you have mitosis. So hair matrix is where you have mitosis that allows the hair to grow. As the hair grows, basically the rest of the hair becomes dead tissue, and the only part that is alive is the hair matrix. Hence, if you are doing a genetic test on somebody, you do need the actual hair matrix, the bulb of the hair, to be able to get a sample of DNA. If you just grab the top parts, those are just dead cells with no DNA in them. So basically no point of it. Erectile pili muscle. These are basically the muscles that are at the base of your hair. And I don't know if I actually have a picture on the next slide. Oops. Sorry, I gotta go back. Sorry, I thought I had another copy of the picture, but I don't. So if you look at here is your hair follicle. Do you see this muscle, this tiny muscle at the base of your hair? This is called the erectile pili muscle. So when this muscle contracts, it literally pulls the hair and make it stand straight. So when you get goosebumps, that's basically the job of the erectile pili muscle. That's basically pulling the hair root and making it stand straight. Which I think is kind of cool. So cats have a really strong erectile pili muscle. But human, not so much, because the hairs are also much smaller. Okay, in terms of the type of hair we have, I think this is really important for you guys to know. We have two types of hair, what we call vellus hair versus terminal hair. So vellus is basically this kind of like baby hair. A lot of people refer to them as literally baby hair. It covers the surface of our body. It is present in both children and adults. And in many cases, it does go away as a person ages. Terminal hair is basically coarse and long hair. This is the hair that you can find as your regular hair, the hair in your eyebrows, your axillary region, like pubic region, the armpit region when you reach puberty. And then for male, when you're thinking about their beard and mustache, that's also identified as terminal hair. Really cool fact that I find it fascinating is that basically everybody starts as vellus hair, then they go to terminal hair, and then a lot of people as they age, they revert back to the vellus hair. So their hair become fine again as they age. And we're not talking about you aging like in their 60s. We're talking about people that are in their 80s, 90s or higher. So the hair goes through that kind of change as well. Alopecia. Alopecia is a very common type of hair thinning that is typically seen after the age of 40. It can be observed in both cases, both male and female. If you ever watch those hair commercials, we have something called true or frank baldness. This is associated with what we call male pattern baldness, which basically means it affects male more than females because it is a X-linked gene, which means it has to come from the mother and then it gets passed on to male. There is a response to a medication called DHT, which is a typical medication you see in, what is that, Rogaine? The ones as for the hair loss. So this is what happens to the hair follicle when there is hair loss even before 40. It depends on the individual. But in many cases, and I don't want to go into too much detail about this, the hair follicle undergoes 3 phases. It basically allows the hair to grow, then that hair follicle dies or kind of goes into, I guess, a sleep mode, and then it comes back and then regenerates and it starts to regrow the hair. So those are the 3 phases that it goes. So if you even look at your scalp, not every hair follicle is active. So it doesn't have an active hair matrix. In people who have hair loss in age of before that age, there's 2 main reasons, or 2 main reasons that I'm aware of. One is that the cycle has sped up rapidly, so they don't really get the time to grow the hair and then drop the hair. So the hair kind of goes grow, die, start to grow, and then repeat that process much faster than it would have happened in a normal individual that would take years. And the other thing is that that hair follicle just failed to produce a hair. So it depends. It literally depends on the thing. Sometimes you actually have medication that significantly affect the hair loss, especially when you think about chemotherapy medication, that has a significant effect. There are other medication. Trauma sometimes causes psychosomatic problems that can cause hair loss too. So really, you can't really say. You have to go see a doctor and try to figure out what the reason is. It's like you have a headache. To me, it's like you have a headache. What could cause the headache? 100 other things can cause headaches. So it's not as simple. Okay, next thing you need to know is nails. So nails, again, we're going to keep it simple. It still does contain cells that have significant amount of keratin. So structurally, again, similar to your hair and your skin. The nail bed, we have what we call nail bed, which is the epidermis that is underneath the keratinized nail plate. So here is my nail bed, the little pink layer that you see underneath. So if anybody has had their nail fall off, the pinkish, nice pinkish skin that you see underneath it. It's called the nail bed. Nail matrix is right here underneath your skin. As the word matrix implies, it's basically the region where you actively have mitosis taking place and your cells are undergoing divisions. And then, you know, the little cuticles that are sticking out on the side of your skin, that extra piece of skin on the side of your nails are called epinicium, or as I said, cuticle. And that's basically just extra skin on the side of your nail. Simple, guys. Don't make it more complicated than what it is. So sweat glands. We actually have two types of sweat glands, which is really cool. I want to spend a couple of minutes on this. We have sweat glands that are called eccrine sweat glands and sweat glands that are called apocrine sweat glands. So eccrine sweat glands are the ones that are essential for helping us cool down. So when you go in a hot sun and you start to sweat, the sweat that you're producing is coming from eccrine sweat glands, okay? This type of sweat, this is really important, guys. This type of sweat is 99% water. It has salts, vitamins, antibodies, and things that can help you kill bacteria. So no protein is found inside this sweat gland. The main goal is to help you basically thermoregulate, so maintain your body temperature. Lots and lots of it is found at the palm of your hand, the sole of, abundant in the palms of your hand, the soles of your foot, and then your forehead. So these are the common places you have higher concentration, but of course you have them throughout your body. The second type that we have, the apocrine sweat glands, is the one that we associate with you being nervous. That's the one that creates the sweatiest smell. So this type of sweat glands, the apocrine sweat glands, are limited to the axillary region, your armpits, and the genitalia area. So the main difference between this sweat and the sweat that is produced for the thermoregulation purpose is that its color is more yellowish, because the main reason is because it has fatty substances and proteins inside of it. So it has all the other components that we listed, plus fatty substances and proteins. So when you release this sweat to the surface of your skin, the bacteria that are sitting on the surface of your skin start to break down your sweat. And as a result, the byproduct of that create the body odor. So your original sweat is not bad smell. What bacteria does to it is what makes the sweat smell bad. Now, this type of sweat gland, apocrine sweat glands, are not active until the person reaches puberty. Eccrine is always active, because even babies sweat if you put them in a very warm environment. So that's really important for you guys to distinguish them. There's usually at least 1 or 2 questions on the exam about this. Modified apocrine glands, we have 2 types of them. Seminiferous glands, which are basically the ones that are found in your ear canal. They produce your earwax. And believe it or not, mammary glands, the glands that are found in the breast that secrete milk, are also apocrine sweat glands that have been modified. Yes, basically. So yeah, and again, look at the locations of these sweat glands. Sweat glands are found in axillary and genital area. So usually when a person is anxious or excited is when the apocrine sweat glands start to release the sweat. A lot of people also say that the apocrine sweat gland also releases pheromones, just FYI. Sorry guys, we're almost done, so bear with me for a few minutes. I know we're gonna go over a little bit. So oil glands is another one that you guys need to know. The location of the oil gland, it's actually built in at the base of your hair follicle. The oil glands are usually not active during childhood, but they do get stimulated when you reach puberty. They produce what we call sebum, which is basically the oil that gets released to the skin. Now, if the opening to the oil glands is blocked, then we get what we call whiteheads. So basically your zits, right? If you don't touch that whitehead and it stays inside your body, it undergoes a process called oxidation, and that whitehead becomes a blackhead. So there is no infection when you have a whitehead, guys. So if you just leave it by, it's just a blocked thing, you can exfoliate, clean it up, and you're good to go. Acne, on the other hand, is when these whiteheads that you have, because it's full of oil, right? Oil is a really good food for bacteria. So when the bacteria get access to this, they start to eat the oil and cause infection in your skin. And that's what acne is. And of course, there's a lot of good medication nowadays in the market that helps control the inflammation. But please distinguish between whiteheads, blackheads, and what is acne. All right, these ones, we're gonna just talk about burns and the three types of skin cancer. So basal cell carcinoma is, there's three types of skin cancer, sorry. Basal cell carcinoma, squamous cell carcinoma, and melanoma. The main reason is frequent irritation to your skin and exposure to UV rays, as we mentioned earlier. When we talk about the skin cancer, guys, two things to remember. First of all, if you're using the word metastasize implies that it is rapidly spreading to the rest of the body, or it has a spread to the rest of the body. And then the second thing is the treatment, if they are sensitive to treatment or not. So basal cell carcinoma, most common type of cancer you see in people who have fair skin tones. These individuals have damage to their stratum basal cells. The reason you see this is because of a high exposure to UV rays. So they spend a lot of time in the sun. Really good fact about this type of cancer. It is the least malignant, which means it's not gonna cause a significant damage. It doesn't metastasize, or it metastasizes very slowly. So it takes years for it to become something much worse. It looks like little pink domes. And if you are one of those people that who have this cancer, literally all you go is you go to your doctor's office pokes it out, does local anesthetics, pokes it out, and then put a bandage on it and you're good to go. No medication, nothing major that you need to know about it. Well, there's like a little cream that you put on it, antibiotic cream, but that's about it. Getting to a more dangerous waters, you get a squamous cell carcinoma, which kind of looks scary too, right? So these are the second most common type, and they can metastasize if you don't remove them. The layer they're affecting is the stratum spinosum layer, which is the second layer from the bottom, sorry. They look kind of red and scaly looking. And the danger about this cancer is, again, because it can metastasize. However, if you do get to it soon enough, you can do radiation therapy and you can surgically remove it. So the chance of survival is still pretty high for people with this type of cancer. The one that everybody needs to be afraid of, regardless if you're fair tone or not, this has nothing to do with their skin tone, is melanoma, which, as the name implies, is going to target your melanocytes. It is randomly starts. It doesn't really affect a certain number of population. Even though people who are white skin or fair skin, they're more susceptible to it, but it can affect everybody. Worst things about this cancer, making it worse thing, highly metastatic. It can spread really rapidly and it's resistant to chemotherapy. So that's really bad. So basically no, very limited treatment, and it can spread to the rest of the body. What I tell my students always, if you start to see a mold on your body, follow this ABCD rule. Look at it. Is it asymmetric? If it is, there's a good chance that might be something dangerous. Does it have border irregularity? So does it go up and down? Does it have that not uniform shape like a mold? Color, if the colors are darker in the color, black, brown, tan, red, sometimes even bluish tinge to it. And the diameter, if it is larger than the size of 6 millimeters, which is the size of a pencil eraser. So be careful, guys. Monitor yourself, your partner, your kids for this. It really has nothing to do with skin tone. It can randomly start, and everybody is in danger. And I know somebody that had this and they couldn't recover from it because it metastasized to the other parts of the body. So take it seriously. If you see a mole growing on your body, take it seriously. Okay, last but not least, we're gonna talk about burns. The main chance or danger about the burns is that it basically can cause significant damage to your proteins. It can cause dehydration in the body. It can damage the tissues, destroy cells. Main reasons for burns are heat, which is the logical one, electricity, and radiation, and sometimes chemicals. Not all chemicals cause burns, but some chemicals do. To evaluate a burn, we use what we call nine rules. Rules of nine, I guess I should say, not rules of nine. Here's a picture, it kind of makes sense. So if you look at the whole body, there's a total percent, 100% for your whole body. So your head, the front is 4.5%, the back is 4.5%. That's 9%. Each of your arms, front and back, are 9%. Your trunk together is 9% on the top, 9% on the bottom. Each of your legs are 9%. Front 9%, back 9%. And then the region for the genitalia is considered 1%. So if you add up all of these, you get up to basically 100%. So that's where the number comes from. But understand this, guys. It's not only what is the percentage of the body that's burned, but what type of burn you have as well. So what does that mean? If you have a first-degree burn, what are the symptoms? First of all, you're only damaging the epidermal layer, which is the top layer of the skin. A lot of it is dead skin cells, right? So what you have is localized redness, you have swelling, you have, of course, pain, but can't really do anything. It's gonna heal on itself. You can just put a little bit of burn cream on it and you're good to go, okay? Typical type of first-degree burn, sitting in the sun for too long, okay? So that's basically a first-degree burn. So are you gonna be okay if you get 100% first-degree burn on your body? Yes or no? If you get 100% burn, first degree in a body, are you gonna be okay? Answer is yes, right? Because it's a first degree. The only thing you damage is the epidermis, which is mostly dead tissue anyway. So that 100% has no meaning, right? Because the damage is very superficial. Now we get to the second degree burn. Second degree burn is you burning through the epidermis and then the top part of the dermis. So you're going to a little bit more deep, I guess. So you see in this case, blisters. Blisters are a really common example that you have a second degree burn. Again, is it as worrisome as a third degree burn, which is the next type of burn? No, because in most cases, this type of burn can heal itself as well. So you're still pretty good. So even if a part of your body has a second degree burn, the chances of recovery is pretty good. Third degree burn is you've gone through both the epidermis and the dermis layer. It's called full thickness burn. So you basically burn through your whole skin, guys. This is the tissue appears as very gray, white, cherry red, or blackened, depending on the tissue damage. This tissue has no edema, guys, no swelling, because all the water basically evaporated. There is no water left. And then the other thing is these people, when you have third-degree burn, at the beginning stages, they don't have any pain because the nerve endings are all gone. However, if they survive this burn, it's going to be extremely painful for these individuals, because as the nerve ending starts to come back, they're going to feel that. They're going to feel the damage to the tissue, and they're going to feel really bad. So a lot of people, if they have third-degree burns in their body and they're surviving that burn, they actually put them in induced comas to make sure they can survive. I know it's really painful. The best way to treat it is they do a skin grafting from a different part of the body and put it on that. There are other technique as well. They remove the dead skin and then do a skin grafting from either yourself or a cadaver to help the skin regenerate itself. But again, it's never gonna be the same way as it was. So usually what they do, if you're looking at the burns are considered critical if more than 25% of the body has second-degree burns. 10% of the body has third-degree burns, which is really, really bad. And then face and hands and feet having third-degree burns. The really high chance of, another thing that you need to know for burns is when you damage the tissue, both epidermis and dermis, your chance of getting infection is severely increased because you have removed that layer of protection, basically. Again, best treatment would be debridement, remove the dead skin cells or remove the dead skin generally. Antibiotics, because again, you're exposed to a lot of things that are outside. Temporarily covering, so they always cover the burn tissue, and then skin grafts. It's really important, if you guys have ever been to a burn unit, which I hope you never have to, they kind of look like those, head to toe covers. Nothing is showing because there's a chance of, spreading the infection. It's kind of like NICU, almost like NICU when you go to NICU and you have really tiny babies. It is really one of the hardest places that you can work at, the burn unit. So I'm glad there are people that actually are strong enough to work with them, but it is extremely difficult to deal with. So, okay, believe it or not, I'm done. Sorry. I know, I thank you guys for sticking with me another extra 13 minutes to finish the lecture, but now you all have the recording. So